First of all, hi everyone, and thank you for joining today. This is the first junior seminar we have in a while. And well, thank you for attending. It's nice to see you here. I'm Alberto Rodriguez, and I'll be the host for the first 4 sessions, and then Clara will take over. And well, as you saw in the schedule we sent, each speaker will have 30 minutes, 25 for giving the talk, and then 5 for questions. And well, on Friday we saw that people were unmuting themselves for asking questions, and I think it was working pretty well. But and you can do that, or if you prefer, you can also post questions in the chat, and we can read them out loud. Well, all that said, I'll introduce our first speaker, Bruno. Bruno Cuevas, who was also organizing this, by the way. Well, Bruno studied biotechnology in the Technical University of Madrid, where he specialized in computational biology. Then he did his bachelor thesis on epidemiological models under the supervision of Fernando García Arenal here in the CPGP. And he later moved to Barcelona for doing a master's on mathematical modeling at the Universitat Autònoma de Barcelona, where he also specialized in complex systems. And he did his master thesis in the Barcelona Supercomputing Center on predicting the stabilizing mutations in protein complexes. And he's now doing a PhD under the supervision of Luis Fernando Pacios. And while also during his PhD, he spent some time in Uppsala University working on enzymatic catalysis. And well, hello, Bruno, and over to you.
Hey, thank you, Alvaro. Okay, let me start. Well, first of all, I'm flattered to start this junior seminar day. I've always been organizing, and also pleased to have this chance to introduce my topic of research, which, by the way, you'll see is not really bioinformatics or computational biology, but it's more related with computational chemistry. I guess that most of you is unfamiliar with this topic. It's a little bit off topic, yeah. So I will spend around half of the presentation introducing why you should care about computational chemistry, and just then I'll start talking about my results and my research. So I always like to start presentations by quoting someone, some major person. So in this case, I couldn't help but quoting Mr. Richard Feynman, Nobel Prize awarded. So in his physics lectures, he asks students what sentence will they give to future generations so they can rebuild science from scratch. And he decided to give them these future generations the atomic theory, that everything is made of atoms that interact, that they are squeezed against each other, etc. So he thought that by providing that sentence, they could figure almost all the modern physics. So in computational chemistry, we decided to listen to Mr. Richard Feynman and to explain the world by simulating how atoms and molecules behave. For that, we use computers. So how can we do it? Well, if you have a computer, the best way to do it will be to just have a program where you input some data and it predicts the movement of all components of chemistry so you can simulate all phenomena. But here we have two problems. First, we have a problem that is common in all branches of physics, which is that we don't know the solution to the equations of motion. But anyway, we can sort this out by making some simulations, but moving in small steps. But then we have a second problem that is more specific of our discipline, which is that we are dealing with electrons. Molecules and atoms are made of electrons, and electrons behave according to the quantum mechanics laws, which makes them quite difficult to model. So when we do computational chemistry, we can use different frameworks. I try to depict them here in this triangle. First, we have the quantum mechanics framework, which is the way we should do things, right? Because at the end is the only one that allows us to model how electrons behave. So to work in quantum mechanics, we need to solve the quantum mechanics equations. for electrons, but we cannot do it exactly, so we have to make some approximations. Quantum mechanics at the end, it has two main shortcomings. First is that its computational expense is huge, so we can only apply it to very small systems. And second, that its accuracy is not as good as we will need to replace lab work. This is why we still do lab work, right? Otherwise, we will just make simulations. Then we have classical mechanics. It's my classical mechanics counterpart, where what we do is we forget about electrons and we just replace we just place some empirical laws that account for the effect of interacting electrons. So then we can simulate our systems just by solving Newtonian mechanics. So we can make simulations of systems containing millions of atoms. So this is the kind of tool we use in these molecular dynamic simulations we do in our lab. Well, it's pretty common in biochemistry and so. But the problem is that it's not very accurate, and it has also another problem is that it cannot describe some specific phenomena, like for instance chemical reactions. Then we have machine learning that is kind of new, and it's pretty interesting right now. There is a lot of hype around it. In machine learning, what we do is we train some mathematical model to reproduce data that we want to know about the systems from data that we already know for our systems. For instance, we can train a mathematical model to reproduce the the energy upon some coordinates of the atoms. Machine learning is, as I say, there's a lot of hype around it. It's providing very good models with almost a computational overhead of classical mechanics. But however, it hasn't been adopted yet by the computational chemistry community. One of the reasons is that communities, they always move slowly. But second is, more important, is that it behaves as some sort of black box. We never know where our results come from. And we never know when they are good or they are bad. So it's still, we can see that in this triangle, we still lack something in the middle that has all the good properties and none of the bias. So this is where I want to place my research. This is what I'm trying to, I want to place something in the middle. But before introducing my research, I'd like to talk about the main character of this research, which is the electron density. As I say, electrons behave according to the quantum mechanics laws, because they are both wave and particles, and they also belong to a family of particles we call fermions that have some specific rules about how they interact with each other, exactly that they cannot share their location and spin. Therefore, two these features make that electrons don't create well-defined orbits around nuclei, but they create some sort of smooth clouds around nuclei that we call electron density. These clouds of charge, what they represent more or less is like the probability of finding an electron in a region of space. So here I depicted the ibuprofen molecule and the electron density for that molecule. So as you see, when we have all these surfaces represent a set of points that share the same value of electron density. So for high electron density values, you can see that our contours are pretty tightly fit to the shape of the nuclei of the molecule. But as long as we reduce the electron density threshold, our surfaces lie every time farther from the nuclei. So the electron density at the end is some sort of soft cloud that decays exponentially according to distance. We like electron density because it's a hub of properties. First is a fundamental property. It's the starting point for density functional theory, which is the theory that is the main tool in computational chemistry to work with, to solve the wave functional problems. Exactly, most of the work, of what is done in solid state or materials is done using the density functional theory. Then we have that it's an observable. It's something we can interact with through experiments. For instance, X-ray diffraction experiments provide us a direct map of the electron density of molecules that we can use to reveal the stuff like, for instance, the protein structure. And it's also interpretable. It means that we have well, as you saw before, we can just depict the electron density and we can make a guess of the shape of the molecule. So that's quite useful. But it's also important in the sense that some theories like atomic molecule theory allow us to explain some chemical concepts that are not very well defined by using electron density, as for instance, chemical bonding. So the goal of my research is to develop a scalable methodology to describe electron density in biomolecules. Biomolecules, they have a problem is that they are generally quite large. We are talking about proteins that have thousands of atoms. So to do that, well, we could have tried to do it from a quantum mechanics approximation, right? But that's kind of a dead end. It's very complicated and nobody has made any major improvements in the last 30 years. So we decided to go to use this machine learning, brand new machine learning tools that everybody's using to pursue our goal. So one of the most important points of this research is that we can obtain electron density, theoretical electron densities from wave function calculations. So when we have a molecule, we can try to solve the motion of the electrons of this molecule by solving the Schrödinger equation in an approximated way using some different methods like DFT, density functional theory, or coupled cluster methodology, or whatever. However, those methods, they are only approximations and they are very expensive. Once we get the result, we get the wave function, which is a very large mathematical object with many terms. So to build the electron density, we need to square that wave function. So it will give us, according to the Copenhagen interpretation of quantum mechanics, the likelihood of finding a particle, right? The problem of doing that is that given that the wave function is already a complex mathematical object and we are squaring that mathematical object, we get a huge mathematical object with many terms that is very difficult to work with, especially if we are working in large systems like a protein. So our approach is to keep the computational complexity as low as possible by using a linear model of electron density, which means that our model will be just a lot of terms multiplied by a coefficient and sum all over. So the number of terms that we are using grows only linearly with the size of the system. Our starting point to do that is the AMD model that we developed in the 1993, where some exponential functions are fitted in isolated atoms to reproduce potentials at the quantum mechanics and correlated limit for those isolated atoms. So as you can see, we can reproduce, this model is very good because it allows us to reproduce almost all properties of atoms with high accuracy by using only a few exponential functions, 3 in the case of carbon, for instance. However, it's not targeted for molecules. In molecules, things are more complex because in molecules we have, for instance, chemical bonding, right? So atoms share some electrons, and sharing those electrons means that we have some directions of space that have more density than others. So this is what we call anisotropy, and it's something that we lack in this model. So we modified our model to provide anisotropy. To do so, we look for analytic form that keeps variables separated, so we can work out mathematical expressions from this in a simple way. So we try different angular functions. We still don't have a very close idea about which one is the best. Here I'm showing you my 2 favorite ones. First, you have the angular Gaussian approximation. This one is very handy from the practical point of view, but from the mathematical point of view, it's very painful. It's to derive anything from it, so we are not using it any longer. And then we have our spherical harmonics description of anisotropy that has some nice properties also, and it's much easier to derive from a mathematical point of view. So once we have anisotropic functions, we can build a whole collection for each of the molecules, and we can multiply those functions by different coefficients that we can optimize to reproduce the electron density of each molecule. So the next question is how we fit those coefficients. And we decided to go for the simplest approach, which is linear regression. We made some from QM calculations. We obtained the electron density, we sampled those electron densities, and then we fit our model to reproduce that electron density. And to do so, we just apply this fancy linear regression. You see here, I mean, it has many terms, but at the end, it's basically a linear regression. So we got these results. The results are kind of good. Here you see in the back and the right that we have the ibuprofen molecule I showed you before. In blue, we have all the isosurface that we predict. And in black, we have the isosurface of quantum mechanics. And as you see, they are pretty tightly fit. So it means that our model is good. And on the left, you see that we have a scatter plot of our prediction of density in random sampled points against the prediction on against the values of reference of quantum mechanics. As you see, we have only some small discrepancies at the level of the very low density area. So this is a good result. Here we have more electron densities. As you see in red, we have our predictions, and in green we have the quantum mechanics reference. As you see, they overlap pretty well. So this is good news. But however, this is not really useful. If we need to get an electron density, a QM electron density to fit our model, then it means that we already have the quantum mechanics reference, so we are not getting anything from it. And I'll have to say also that our model is obviously not so accurate as the reference density because it's not so complex. So how can we turn this useful? Well, here's where we decided to use machine learning, so we can skip the quantum mechanics calculation and we can give those coefficients I showed you before directly the value they deserve. So for doing that, we developed this methodology we call H2MDNet, which is a neural network based methodology with some modifications for this specific problem. So what we will do is in this prediction model is that we feed our model with things we already know for a molecule, like coordinates, atomic numbers, molecular topology, which means bonds, a molecular charge, and we will produce from that the electron density. How we do it? Well, we have that coordinates and atomic numbers are converted to some what we call symmetry functions. These symmetry functions are a way to show molecular data without in a roto-translationally invariant way. And then we feed the neural networks with these symmetry functions, and we produce each of the different coefficients of our model. Then we just normalize all those coefficients so the charge of the model gets preserved. And finally, we reproduce electron density. To reproduce this model, to train this model, we made around 50K high-level quantum mechanics calculations on small molecules. And we tried to train the model on those densities. But instead of going directly to the densities, we made an intermediate approach where we fitted our linear models using the least square regression I showed you before to those electron densities, and then we train our network to reproduce those coefficients. So we are training against the result of the fitting. So here you see the results for an FDA-approved molecule benchmark, which means drugs. Drugs are quite complex generally. So here you can see that our predictions of the coefficients are quite correlated, although we have some small errors, but those are not really important because the model is quite robust. And we also get some unexpected outcomes, like for instance, that we can derive atomic charges from our predictions with very high accuracy, as you see in the lower right corner. Moreover, we have that our scaling is pretty good. It remains almost linear. Here we are comparing the scaling of our tool on the lower part against the scaling of the quantum mechanics tools that we use to get the electron densities. And as you see, quantum mechanics here in this case is scaled as a power of 4, which means that a calculation on a system that is twice larger will take 16 more time. So this is a pretty good result that is quite common in machine learning. However, this is not over yet. I mean, we haven't solved the problems of quantum mechanics, as you can guess, because at the end, what we need is to improve accuracy. Here I showed you the mean square error of different methodologies against the best quantum mechanics reference we can get. And as you see, well, we are better than some sloppy quantum mechanic methods or some empirical or some very small basis set calculations. But we want to get better than, or at least as good as the quantum mechanic methods that are used in the real world. So we are we're working on the next release of the this neural network, what we call A3MD-Net. This one will have some changes. For instance, it will learn directly from electron density. It will use whole numerical integration. We will use a slightly different density model, but more importantly, you will use a different approach for the machine learning part. We will be using what we call message passing neural networks, where let's say that atoms talk with the atoms around them, so they can figure out the atomic environment they have around them, and so they can produce then the results for the coefficients for the electron density model. And finally, I didn't want to finish my presentation without showing you that our electron density models can be already used to reproduce electron density in large biomolecules, like for instance, this tryptophan cage. We still haven't developed an application for this on large molecules. I hope we will someday in the future, but well, as you see, we are working more in the fundamental part of this theory. This is all. I want to thank you all for the presentation. I see that I made it quite quicker than my previous attempt, so sorry if I was too fast. And I want to say bye by thanking the people around me, especially Luis, Luis Fernandez Pacios, who is my thesis director, and also all my group, which they work in a different field, but they always make the effort to understand what I say and what I do. So they are lovely people, and I want to thank them. And that's all. Thank you.
Thank you, Bruno. Are there any questions?
I have one question, Bruno. So I don't see the hand to ask for my turn. So could I ask the question?
Sure.
Okay. So, well, first, congrats for this progress. And I have a question regarding, so the majority of the data, so based on this comparative analysis that you have done in one of the last slides. So do you get that, and also based on your last slide, that it will be feasible to evolve the method to your original objective of approach this problem with small proteins, or still you see this is far away?
Well, we are. The problem here is that, of course, you cannot be as good as quantum mechanics because it's way more complex, right? So in that sense, maybe I said that we want to get as good as quantum mechanics, and it's not true. We want to get to some point where people can use us as a cheap alternative to quantum mechanics. And to do so, we think that it's quite feasible in the future. We are adding every time more accuracy on the network. We are training lately on small proteins, which is a good starting point to reproduce electron density on proteins. And we are even getting to make some energy calculations, DFT energy calculations. So I hope that it's not that we are going to replace quantum mechanics. I mean, if someone working quantum mechanics will be hearing me now, he will say, okay, this guy is pretending too much. But we think that we can get to be a cheap alternative, something that you can run in your laptop to get results that you will need a supercomputer to get. And of course, not with such accuracy, but okay, as I say, if you want accuracy, go to expensive quantum mechanics. Right now, almost it's very difficult to apply quantum mechanics in proteins. So this is like an alternative to nothing. So we think that is very good.
Okay, that was also my other question is that you already answered regarding the potential of quantum mechanics in protein ligand determination, but you already answered my question. Thanks.
Okay. Well, I think I haven't talked about too much about the applications regarding proteins and ligands, but one of the things we are working, for instance, is in making some sort of quantum mechanics docking. So we can dock ligands and proteins using not quantum mechanics, but expressions where electrons are explicit. So that's really a large improvement.
More questions?
Mm-hmm.
Well, I actually have one question, and well, I don't know if it's a silly one, but
there are no silly questions. So,
what's the application of quantum computing on this field? Are we going to be able to do quantum mechanics on these computers on large molecules, or is this not feasible at all?
Of quantum computing?
Yes.
Okay, the objective of quantum computing is to let all computational scientists with unemployed in the short term. But in this case, I guess that there are already some applications of quantum computing in neural networks, and they look great as everything in quantum computing. The only problem is we don't ever know when it's going to arrive. But yeah, at the end is that it provides larger computational complexity. I mean, it allows your computers to tackle problems that are unfeasible right now. So maybe when we have computational quantum computing, like for real, then it will not be necessary to use my machine learning approach. But anyway, until then, well, I guess that we have still something to do. And after that, I guess that we will try to do all the work, like, I don't know. Yeah.
Okay, thank you.
Okay.
Bruno, I have another question just before your time is close to you. So in this new E2MD net methods, the computational, I mean, the power of the computational requirement is, I understand, is reduced compared to the other type of quantum mechanic calculation, correct?
Yeah. Yeah, it's reduced in many terms. Of course, we are not getting so much information as a quantum mechanics. It wouldn't be fair to say that we are getting the same output as quantum mechanics, because it does a lot of stuff that we don't do right now. But yeah, it's like we are taking, I don't know, 10,000 times less time than. So yeah, it's pretty.
And you are mainly use GPUs or other type of?
Yeah, everything we are doing is in GPU, and GPUs are very good for this, because at the end, most of the operations we are doing can be run massive parallel arrays. So using a GPU allows you, for instance, to train your neural networks in one or two orders of magnitude less time. And they are being also very handy for making the numerical integrations that we are doing right now. Because at the end, it's like you have, for each density, you have to integrate, I don't know, 10,000 points. And if you do it on a GPU, then you can do everything at the same time. So yeah, GPUs are key on this development.
Okay, thanks.
Okay, thank you, Bruno. I think we don't have time for more questions. So we'll just go our next speaker, who is Diego Rebaque Moran, who studied biology and did a master's on research in fundamental biology and biomedicine in Universidad de León. And he specialized in plant cell during his final master project. And now he is developing an industrial doctorate in collaboration between Antonio Molina's lab and Plant Response. So thank you, Diego, and over to you.
Okay. First of all, thank you, Bruno, for sharing your work with us, and thank you to the Junior Seminar Committee for organize this seminar session. This project is part of an industrial doctorate, as Alberto said, in a collaboration between Antonio Molina's lab and the company Plan Response Incorporation, that is a joint venture from CBP's spin-off. This project came to give a solution to a problem, that is that around 30% of crop production is lost due to plant disease. We need to reduce the pathogen impact in crops in a sustainable way without the use of pesticides and other chemicals with a high harmful impact to the environment. Our objective is the obtaining of new products, called biostimulant, the civilian in the image, derived from immune sense elicitors to enhance crop protection. Well, when a pathogen interacts with the plant, they find a first physical barrier, that is the polymeric cell wall, and they have a wide set of tools to break through this barrier. In this cell wall hydrolysis, a lot of molecules are produced, and some of them can be perceived by the plant by the pattern recognition receptor in their plasma membranes, and in consequence, immunity is triggered in the plant. These elicitors are known as damage-associated molecular patterns, but also molecules from the pathogen can be perceived by the plant as microbe-associated molecular patterns. The list of known MAMP and DAMPs are very high. Most of the molecules are peptidic molecules, but also carbohydrate molecules has been described as DAMP or MAMP. For example, the well-known MAMP chitin or galacturonides that were the first cell wall DAMP described. It's very interesting that beta-glucans are present in both lists. Beta-1,3 glucans that were described as trigger immune responses in Arabidopsis by our group, and the beta-1,4 glucans that has been previously, more recently described by others. Glucans are linear changes of glucoses that we can represent as this hexagon, linked by beta-1,4 bond in the case of cellulose-derived oligosaccharide, or by beta-1,3 linkage in the case of laminarin oligosaccharides. But exists another kind of linear glucans that combine these two types of linkage, that are the beta-1,3,1,4 glucans. These glucans introduce a beta-1,3 linkage after a variable number of beta-1,4 linkage. These glucans are also known as mixed linked glucans, or MLG. Where our starting question was, can plants also perceive these glucans? Well, these mixed linked glucans were described as cell wall polymer in Poaceae order group of plants, but more recently has been also described in Echicetaceae family and in other group of organisms. For this project, we depart from 3 different materials: a beta-glucan from barley, hemicellulosic cell wall fraction of Echisetum arvense, and a lichenan from Cetraria islandica, a lichen. We first tried these polymers in apocorine system, that is a transgenic line of Arabidopsis that allows us to measure calcium influxes triggered by elicitors. And when we compare the treatment with the well-known chitohexaose derived from chitin, we obtain no signal because these polymers have a high degree of polymerization that avoid them to go through the cell wall and be sensed by the receptor in plasma membrane of plants. Then we treated these polymers with an enzyme called likenase that is specific to these beta-1,3,1,4 glucans that hydrolyze beta-1,4 linkage in the reducing end of glucoses that possess a beta-1,3 linkage. We didn't use cellulases that are able to hydrolyze this polymer 2 because cellulases randomly hydrolyze beta-1,4 linkage and are able to produce a sequence of beta-1,4 oligosaccharide that has been described as active, but we are not interested on them. So with this likeness, the digestion, we obtained a mixture of MLG oligosaccharide that when are dried in apocrine system, we obtain these peaks of luminescence corresponding with those calcium influxes. We analyzed this digestion product by size exclusion chromatography, and the illusion obtained were analyzed by thin layer chromatography to analyze the composition of the peaks of the illusion profile. If we overlap in the results of these two chromatographic techniques, we obtain something like this, where we can see that the main peaks of the digestion product are made up of a trimer that we have called MLG43 because it is composed by 3 glucoses linkage by a beta-1,4 linkage and a beta-1,3 linkage. To ensure that this MLG structure that trigger the calcium influxes, we obtained different structures from different sources. These two commercial sources of MLG from Barley and X2 and in collaboration with Fabian Frengle and Prieto Dalla Bernardina, we chemically synthesized MLG structures that are not possible to obtain from the nature. That we obtain is that all the triad structures trigger calcium influxes. Only some of these chemically synthesized MLGs has non-activity or very low, but this is probably due to the presence of this linker as a product of this chemical synthesis. Well, when a plant perceives an elicitor, 4 events appear that can be measured by us as immune-related hallmarks. And early calcium influxes and the production of reactive oxygen species, then a signaling cascade of manganese phosphorylation, and finally immune-related genes are induced. Calcium influxes were analyzed in this apocrine system that I talked about. ROS production were analyzed by its reaction with luminol and peroxidase. Manganese phosphorylation were antibody labeled in Western blot, and induction of immune-related genes were analyzed by real-time quantitative PCR. We obtained a luminescence peak corresponding with calcium influxes, as we saw before, and we have a peak of ROS production with the treatment of MLG43, lower than keto exhaust, but is significant too. This is very interesting because ROS production has not been described in beta-1,3 and beta-1,4 glucans. We obtained manganese phosphorylation in MPK6 and MPK3 with a maximal intensity at 10 minutes, and all the immune-related genes that we analyzed appeared overexpressed in a similar pattern than keto exhaust. To further analyze the changes in the expression of genes after the treatment with MLG43, we performed a transcriptomic analysis. In an RNA-seq analysis, we found 1,299 misregulated genes, most of them upregulated and overlapped with keto exhaust. In a gene ontology term analysis, we obtain a network like this, where we can see that almost all the gene groups are related with defense response, innate immune response, or signal introduction of stimulus and stress, both biotic and abiotic stress. Well, given this similarity in the global gene reprogramming triggered by MLG43 and ketoexaose, we wondered if refractory stage will exist between the application of these two glycans. To check this, we use again the Apochlorine system, but in this case, after application of one of the molecules, we applied the other 10 minutes later. We can see that when you treat the plant with the same molecule in the second application, you obtain no peak because the plant is not able to respond again to the same stimulus. But when you treated the plant with MLG43 after it had been treated with ketoexaose, or in the other way, you treat the plant with ketoexaose after it had been treated with MLG43, you obtain a second peak. It's true that this second peak is not as high as the original one. So this result suggests that the mechanism of perception of these glycans and the pattern recognition receptor that are involved in their perception are not identical. We know that ketoexaose, that is a chitin-derived oligosaccharide, are perceived by SERP1 receptor, and that is a PRR from Lysen family that has a crucial role in the perception of glycans. We know that the perception of ketoexaose depends on LIG4 and LIG5 AFCO receptors. So we tried MLG43 in two mutants, a SERP1 mutant and a double mutant for LIG4 and LIG5. And you can see that the treatment with ketoexaose that produce these bands in wild-type cold plants disappear in the mutants. And when we treated the mutant with MLG43, in spite of the intensity at 5 minutes is the same, at 10 and 20 minutes, the intensity is lower. So SERP1, LIG4, and LIG5 are partially involved in the perception of MLG43, but it might be as a co-receptor. Well, with all of these results, we can say that beta-1314 glucans trigger immune responses in plants, but that we need are molecules that enhance the resistance in plants against pathogen. To check if MLG43 are able to do this, we treated Arabidopsis plant with a solution of MLG43, and 2 days later, we sprayed them with aconidia suspension of the biotrophic omised Jaloperonospora arabidopsis. 7 days later, we count the conidia per milligram of plant, and that we obtained was that the presence of Jaloperonospora in plants was lower in those plants that had been previously treated with MLG43. This is very interesting and give us a question, that is how MLGs are involved in the Arabidopsis Jaloperonospora arabidopsis perception, because until now MLGs has not been described in the Arabidopsis cell wall. So maybe MLGs are present in the Jaloperonospora cell wall. We wanted to quantify the MLG present in Jaloperonospora arabidopsis cell wall, but there is a problem. This organism is a biotroph, so its hypha grows between plant cells. So it's impossible to separate the omised material from the plant material. So with this aim, that we did was to wash infected plant to obtain a mixture material of plant and hypha and a conidia suspension. We quantified the MLG present in this material, and we obtained that in uninfected plants, the amount of MLG is under the detection limit. So we can say that in the Arabidosis cell wall, there is not MLGs that is described. But in Conidia cell wall and in the cell wall extraction of infected plants, MLGs are present. So in this case, MLGs are part of the pathogen, so are acting as microbe-associated molecular pattern. Well, MLG43 are able to enhance resistance in Arabidosis against yellow peronospora, but Arabidosis is not a crop of agronomic interest. So we tried MLG43 in tomato and pepper plant that are crops of agronomic interest, treating the plant by spray of an MLG43 solution. 48 hours later, we inoculate tomato plants with a suspension of the bacteria Pseudomonas syringae, and in the case of the pepper, we inoculated with a suspension of Sclerotinia sclerotorium or with a suspension of spores of Botrytis cinerea. Some days later, we count the colony-forming units per leaf area in the case of tomato, and we measure the disease symptom indexes in the case of pepper. That we obtained was that the bacterial present in tomato was reduced at 11 days post-infection in those tomato plants that had been treated with MLG43. In the case of pepper, the presence of Sclerotinia sclerotorium was reduced at 9 days post-infection in those plants that had been treated. And in the case of Botrytis, even at 5 days post-infection, we can see significant differences. Well, as I told you in the introduction, these beta-1314 glucans were described in the cell wall of Poaceae and Echinacea family of plants. Into the Poaceae family, there are some species that are of agronomic interest, as wheat, maize, or rice. And we know that MLG oligosaccharide can be released along an infection of these plants, because cellulase is one of these set of tools that pathogen has to break through in the cell wall. And beta-1314 glucan are present in these plants. So what is the potential of MLGs in Poaceae crops? Well, in collaboration with Andrea Sanchez and with Gemma Lopez's help, we treated wheat plants with MLG43 solution, and 24 hours later, we sprayed them with a spore suspension of Thymoseptoria treated. 20 days later, we measured the percentage of leaf coverage by lesion produced by these fungi, and that we obtained was that the impact of the pathogen was very significantly reduced in those plants that had been treated with this oligosaccharide. In addition, we analyzed the expression of some immune-related genes in wheat, and we obtained that all of them appear overexpressed in those plants that have been treated with the MLG43. Well, as an overview, we are able to obtain MLG oligosaccharide by chromatographic technique from the enzymatic digestion of different organic sources. These oligosaccharides trigger immune responses in Arabidosis and are able to enhance the resistance in crop plants to pathogens. All of these results are now patented and collected in a manuscript that has been already accepted, but some lines of study are open from this result. First of all, we want to identify an optimal source to obtain these oligosaccharides, ideally biowaste from other industries. we want to better understand the perception mechanism of Mrg43 in plants. And finally, we want to develop the best formulation for production commercialization to finally transfer this knowledge from laboratory bench to a real crop field. Well, this work is possible due to the collaboration of a lot of people. I have to thank Antonio Molina's lab and Plant Response lab. Here you only have the names of those that have been involved directly in this project. A special thanks to Antonio Molina and Hugo Melida, that are my supervisors. Andrea Sanchez for your collaboration, and thank you to our Plan Response, and especially to those really involved, Jolie, Rosa, and Fred. Thanks to Francisco Villaplana from Kungliga Teknika Högskolan from Stockholm, and to Fabian Friendly and Preto Bernardina from Planck Institute of Quality and Interface from Germany. And of course, thank you all for your attention. Now I will happy to answer your question about this work.
Great, thank you, Diego. So if anyone has questions, please unmute yourself and go ahead.
Hello, can you hear me?
Yes.
Okay, I have one question, and it's about, because with this treatment, you are inducing a response directly from the plant. And this is a very different approach than using pesticides, which are directly trying to kill the pathogen. So in this case, you are asking some response from the plant, which involves a change of gene expression and maybe also production of proteins and so on. And this could happen during different parts of the plant life cycle also. So in general, my question is, what do you think that this could have some effect on, or some influence on crop yield at the end of the crop life cycle?
This is one thing that we want to further analyze to better understand how plants react, what is the mechanism that is behind this response of the plants triggered by this Mrg43. And we have not analyzed if it could have some effect, for example, in plant growth or others. It's very interesting. It's something that is open from this result. But no, we have not analyzed what is the specific response that is triggered in the plant by this Mrg43 that allowed them to enhance their resistance against the pathogens.
I think, thank you. I think it would be very interesting to check whether it has some effect on yield, because, for example, if you have one crop which is producing, I don't know, X tons per year of grain, for example, barley or wheat, but then you treat with this and you are maybe reducing the yield, or maybe not, but maybe you're reducing the yield, but in the end you are more secure or more stable against the pathogen. So this could be some kind of trade-off, and also maybe depending on when you apply the product, and you could have a different effect on crop yield. So this would be very important for the product, I guess, and for the people working in the field.
Of course, as this technology is in the way to be applied in crops, this must be an objective from now in the analysis. Thank you.
Thank you very much, and congrats for your work. It's a lot of work and very, very good results.
Thanks.
I think Fernando raised hand.
Yes, I have a couple of questions, Diego. I may have missed something because there was a slide that passed very quickly, but I didn't see that the treatment with this 43 compound had any effect on the resistance of tomato to Pseudomonas syringae. Was it so or not?
Yes, it has an effect.
It has an effect?
Let me show you here.
Yeah. Here's the next slide.
This in tomato against Pseudomonas, right?
Okay, yeah, it's a logarithmic scale on the y-axis. All right.
We
I
obtain
made this.
significant differences at 11 days post-infectum.
Yeah.
The measure of 0 days post-infectum is a control of the.
Yeah. Is not a big effect, but it is some effect. And the other question is related to the Yellow-peronospora experiment in Arabidopsis. You conclude that the mixed glucans can be perceived as signals allowing to detect the omycete, but is this hypothesis necessary? Is not sufficient to explain your results the fact that they are elicitors of the resistance response in the plant?
Well, this was our idea that we know that this molecule is active and trigger the immune response in plants, but we have find other experts in immune responses of plants that wants to classify it. That I agree with you that it will not be needed because it's active. You can classify it as MAMP in this case, but in the case of wheat, you can classify it as DAMP as is present in the cell wall. But well, we perform this analysis because there are other people, other experts that wants to do this classification. So these results show that maybe the classification is not needed, or that we have to have the mind open to like this.
To other possibilities. All right, thank you. And a final comment, I think that for future presentations, you should modify your first slide because plant diseases are not the cause of 30% of losses. Okay, 30% of losses are the results of diseases, pests, and weeds. And diseases by themselves are about 13-15% of losses. Okay, that's the same.
Okay, I will check. Thank you.
They are important, but not. It will be terrible if they were 30%. Okay, thank you, Diego. Very interesting work.
Okay, I think we have time for one more question if anyone wants to do so.
If there is no more question, I think that Loretto is waiting for do his presentation.
Okay, we can just go ahead then with the next talk, and which is going to be our next speaker is going to be Loretto Espinoza, who graduated in biology at the University Autonomous University in Madrid in 2015, when he did her bachelor's project in the Department of Plant Physiology, working with cyanobacteria in Soledad Sanz Alferez's laboratory. And she later did a master's degree in biotechnology, also at Universidad Autónoma de Madrid, where she worked in plant development and the CNV under the direction of Pilar Cuevas. And in 2017, she joined the CVGP to do her PhD in the laboratory of José Antonio Jarillo and Manuel Piñeiro. And now she's going to tell us all about it. So thank you.
Okay, thank you for the presentation. You listen to me?
Yes.
Okay. Well, today I want to show you some of the results that I have obtained during my thesis, which are centered in the study of the role of Arabidopsis for histone acetyltransferase complex subunit EF6 in the control of developmental processes. As sessile organisms, plants can modulate their developmental programs in response to both environmental and endogenous factors, and this provides species with a great plasticity. In this process, it's very important epigenetic factors because they establish gene expression patterns that allows this plant plasticity in this developmental plasticity. Chromatin there are many processes that remodulates the chromatin, and the combination and crosstalk among them contribute to the regulation of many biological processes, including plant development, such as the floral transition. In our lab, we are highly interested in how this process is regulated. The floral transition is the change from the vegetative phase to the reproductive one, and for that is necessary changes in the pseudoapical meristem identity, because during the vegetative phase it produces leaves and branches, and then in the reproductive one it produces flowers. This process is modulated by the floral inductive pathways, which is induced by both endogenous and environmental factors. All these pathways in the end control the floral integrators, where among them it highlights the role of FT, which is maybe the most important one. Chromatin remodeling processes control this process, and they are they control the expression of these genes, and maybe the most paradigmatic example is FT. The floral repressor FLC, not FT. FT is also controlled by these processes, but the most important example is FLC. FLC is controlled by many different chromatin remodeling processes, and one of it, and is one of the most studied one in the last years, is the change of the canonical histone H2A by the histone variant H2A seed. And this process is performed at the Swar1 complex, which is a chromatin remodeling complex that uses the energy from the hydrolysis of ATP to produce this change, and the deposition of this histone variant promotes the expression of this repressor that in the end repress flowering. So Swar1 complex is very important in the regulation of the flowering transition. And for example, mutants from some of the subunits of this complex are all of them early flowering, among other developmental process changes. Swar1 complex was firstly described in yeast, and it's interesting that some of the subunits of this complex are also present in other chromatin remodeling complexes, which is the case of these 4 subunits that are also present in the NUE4. This complex, the NUE4 complex, is a histone acetylase complex that can acetylate histone 4, histone 2, and also the histone variant H2A seed. Both complexes are functionally related, and there is a working model proposed from these 2 chromatin remodeling complex, which consists in that a NUE4 complex acetylates histone 4, and this acetylation recruits Swar1 complex that recognize this acetylation mark, and deposits it produces the exchange of the H2A by H2A seed. And then the NUE4 complex acetylates the new histone variant, and this promotes gene expression. This working model is reinforced by the existence of the TIP60 complex, which is a chromatin remodeling complex composed by subunits of both Swar1 and NUE4 complex, and it is able to also acetylate histones and to deposit H2A seed. The NUE4 complex was firstly described in Saccharomyces cerevisiae, and it is composed by 13 subunits. But you can find it in the big form of the 13 subunit, and also you can find it in a piccolo NUA4 subcomplex, which is composed by the catalytic activity, catalytic subunit ESA1, and also by these 3 proteins, AF1, AF6, EBL1, and ING2. The existence of this complex in plants nowadays remains an open question, since it hasn't been characterized and also isolated in plants. But in Arabidopsis, there are homologs for most of the subunits of this complex, in exception of EF5, and also for most of them are also duplicated. EF6, which is the protein of interest in my thesis, is also codified by 1 gene, And it is interesting that the function of this protein, it remains an open question nowadays, even in other organisms. So it makes this protein as a good candidate to explore. The characterization of different subunits of the putative NUA4 complex in plants have revealed that this putative complex is implicated in many processes, including plant development and also flowering time. So taking all to this in account, the object of my thesis is the molecular and functional characterization of Arabidopsis EF6. For that, we want to perform genetic and molecular characterization of EF. Also, we want to characterize the role of EF in flowering time and other developmental processes. And finally, we want to understand the function of EF6 in modulating the organization of chromatin. All these efforts are centered into understand how EF6 can modulate flowering time as part of a NUA4 complex. So EF6 is highly conserved to species, and we want to determine the degree of similarity among them. So for that, we compare the protein sequence of the EF6 from Saccharomyces cerevisiae and other plant species, and even from Homo sapiens. And we found that there is a highly conserved region that belongs to the NUA4 domain, which is essential for proper function of the protein. Then, if you remember, I say that EF6 is part of the piccolo NUA4 subcomplex in yeast, and we want to check if Arabidopsis EF6 is able to physically interact with other subunits of this piccolo NUA4 complex. And for that, we perform coimmunoprecipitation assays in Nicotiana ventamiana leaves. And until now, we have found that EF6 is able to interact with ING2, which is the homologs, one of the homologs from the ING2 of yeast. And also in collaboration with Javier Barrero and Laura Bouza, we have established that EF6 physically interact with both homologs of EPL1 of yeast. This confirm or is in agreement with the idea of a NUA4 complex in plant, or at least the existence of a piccolo NUA4. Then, in order to characterize the function of this protein, we identify and isolate 3 TDNA insertion lines from the Salk collection, and we measure their expression, the expression of EF6 in these TDNA insertion lines, and we found that EF1 has the same expression levels of Columbia, but the other 2 are mainly null alleles. Given that EF6 is the insertion of EF6 is nearest to the ATG of the gene, we decide to center our efforts and all the experiments in this allele. So as we are interested in characterize the role of this protein in flowering time, we measure the flowering time of the mutants compared to the wild type in long day conditions and also in short days. It is interesting that this mutant doesn't show any alteration in flowering time under long day conditions, as they are able to produce the same amount of leaves of the Columbia before bolting. However, in short day condition, the mutant is clearly late flowering compared to wild types, and even some of the mutants are unable to flower and they die. So we can affirm that EF6 controls flowering time in Arabidopsis, at least in short day conditions. Among other developmental alterations that we observe is that EF6 mutants have a paler color, which is accompanied by a decrease in chlorophyll content. And because of these results, we ask ourselves if the photosynthetic machinery was affected. Because of that, we decide to check the chloroplast ultrastructure with transmission electron microscopy. And we found that in Columbia, the chloroplasts are well aggregated, while in the EF6, the grana are not that doesn't present that aggregation. And also, in general, in EF6 mutants, the area of the chloroplasts are bigger than in Columbia, and this could be maybe because of this less aggregation of the thylakoid. In addition, we observed that EF6 plants display a reduction in rosette diameter and also alteration in leaf state because some of the leaves are curly. So EF6 it is not only regulating flowering time phenotypes, it is also regulating other developmental processes, and also it has a role in chloroplast biogenesis. We want to check the expression patterns of EF6, and for that we analyze the expression of this gene in different tissue samples and other in different developmental moments of plant development. And what we found is that EF6 is expressed at a low levels in all the tissue samples that we analyzed, and this is in agreement with public. With what is published in public databases in the U Toronto. And also as a tissue level, we want to check the expression, and for that we fuse the EF6 promoter to have a beta-glucuronidase reporting gene, and what we found is that it preferably expressed in the vascular tissue of leaves and also in roots, and even it is highly expressed in the upshoot apical meristem. So these expression patterns of EFs that are ubiquitous are in agreement with the idea of EF6 as a regulator of many plant processes, such as the development or of biological processes. Then, given that EF6 only has a flowering time phenotype in short-day conditions, we decided to check the expression of the gene in this photoperiod, and we found that during the light hours, EF6 is expressed while its expression decreased during the night hours, and this could be related with the fact that it only has a phenotype in that photoperiod. Okay, to continue working and trying to study this protein, we developed some biotechnological tools, and we create transgenic expression light that overexpress the EF6 fused to GFP. And in this case, we generate two overexpression lines that we are able to detect by Western blot analysis, and this construct allows us to confirm that EF6 has a nuclear subcellular localization, which is in agreement with EF as part of a chromatin remodeling complex. We also want to generate this transgenic line to confirm that the mutation displayed by the mutants are due to the lack of EF6, and with complementation assays, we confirm that this is true because the transgenic lines are able to complement the flowering time of the mutants, and also they are able to complement the rosette diameter and the chlorophyll content. So we can affirm that the lack of EF6 is the responsible of the observed phenotypes. In the same way, we also generate transgenic light that overexpress EF6 fused to MIG. And in this case, we generate 5 overexpression line, and we were able also to detect by Western blot. And in this case, all the transgenic lines partially rescue the phenotype, because all of them are able to rescue the rosette diameter or the chlorophyll content, but only one of them, which is the 153, is able to totally rescue the late flowering time. So we select this line as a good candidate to continue working with in addition to those fused to GFP. Then we want to check the effect of EF6 in gene expression, in global gene expression, and for that we perform transcriptomic analysis by RNA-seq. In both Columbia and EF6 mutants grown under short-day condition, which is the condition in which we observe the developmental alteration that we are interested in. And what we found is that there are 693 misregulated genes, where 632 of which were upregulated and only 61 were down. And this induces us to think that EF6 is a repressor of the expression, maybe because it's a direct repressor, or maybe because it promotes the expression of repressors. It is interesting that the many genes that are misregulated in our data are regulated by methylation processes, which is the case of FWA, which is one of the most upregulated genes. This gene encodes a protein that physically interacts with EFT, which is one of the promoters of flowering time. So, and this FWA inhibits the function of FT, and this could be a good candidate to explain the late flowering phenotype displayed by the mutants. Also, MAF4 and MAF5, which are flowering repressors, are upregulated in our data, so they are also good candidates. Among the downregulated genes, it highlights CLASI2, which is a chromatin remodeler that that acts as a gene silencing. With these, all the misregulated genes, we perform gene ontology experiments, analysis, with both molecular and biological processes. And among the molecular function, there is an enrichment in genes associated with catalytic activity and DNA binding, which is in agreement with the idea of EF as part of a chromatin remodeling. And regarding to the biological processes, there are many genes associated with cellular processes and molecular, and there are also some genes associated with reproductive processes and developmental processes, which is in agreement with the alteration displayed by the mutants. Then to continue with the characterization, with the functional characterization, we perform genetic approaches that consist in the combination of mutants for EF6 and also mutants for genes that are affecting the flowering time of the plants. And this is an approximation that we performed first, which is the combination of EF2 with JAF9A, which is one of the subunits that are present in both complexes. And we perform analysis in long-day conditions. So due to EF6 only has a phenotype in short day, we have to perform this experiment in that conditions if we want to check that if there is any genetic interaction between both mutants. But if we look to the rosette diameter, we can find that EF62 and JAF9 are smaller than the Columbia, and there is an additively effect in the double mutant. But well, in any case, this is just an approximation to show you how we are going to proceed in the future. So with the results, we can affirm that EF6 physically interact with co-components of the PICO NOA4, ING2, and EPL A and B. Also, we can say that EF6 participates in the activation of flowering time under zootic photoperiods, and also it participates in chloroplast biogenesis. EF6 is broadly expressed and accumulated during the day in short days. And finally, we found that global transcriptomic analysis revealed changes in key genes of flowering time regulation. For the future, we want to analyze the expression of master genes controlling the floral transition in the mutant lines and also in the overexpression generated lines. We want to, as a chromatin remodeling complex, we want to perform tick PCR experiment to quantify the levels of histone H4 acetylation and other histone marks in identified putative direct targets of EF6. And we also want to perform TIC6 approaches to reveal the organization of chromatin at the genome-wide level. We want to continue characterizing the existence of genetic interaction between EF6 and genes that encode regulatory genes that are associated with flowering time regulation. And for that, we are performing cross with EF and genes that are affected, with mutant for genes implicated in flowering time regulation, and also by realizing crosses with EF6 and mutants for both NUA4 and Swar1 complexes. And finally, we want to look for EF6 interaction by in vitro immunoprecipitation assays also because we wanted to confirm that the existence of Piccolo Noir in plants, and we want to perform pull down assays to find new possible interactors with EF. So finally, I want to thanks to all the people of my lab, and especially to Manuel Piñero and José Antonio Jarillo, which are my directors. And also I want to thanks Veronica, because he was working with me during 2 years. And of course, thank you to all of you for your attention. If there is any question.
Thank you, Loretto. Yeah, if anyone has questions, please unmute yourself and go ahead.
I have a question. Well, two questions actually. Regarding the thing that mutants are only affected by short daylight.
Mm-hmm.
So one thing I wanted to ask is like, how did you find this? Is this really common when you analyze flowering, that some genes affect only during short day or only during long day? Or is that you were just trying different conditions and you found that it affects short day flowering?
There are some genes that are associated to photoperiod, because there are genes that are mutants that are affected in this process. Normally are late or early flowering in both conditions, but there are some cases in which the phenotype is only revealed in one of both conditions. So this is not an strange behavior.
Okay. And second, well, but I guess that this is like a more broad question because it will also happen in other proteins. It's like, how do you think is short daylight condition affecting how is this protein interacting with short day conditions?
Sorry, I don't understand.
Yeah, how is this protein, this AF6 protein, interacting with the short day conditions? Is it through some other regulators, or is like
that's what we are going to want to understand.
Okay. Okay, thank you. Very great talk, by the way.
Thank you.
Anyone else wants to ask something?
Loretto, have you checked? I mean, you have generated a lot of mutants and double mutants, but have you checked overexpress online? So just to overexpress, you have a different, I mean, the opposite phenotype, but it's not sufficient just by overexpressing this gene.
We have generated overexpression lines of EF6 in Columbia background, but we haven't characterized them yet.
Okay, thanks.
We still have time for a few more questions, so if anyone.
So in this case, I have another one, Loretto.
Mm-hmm. Yeah.
Antonio Molina. Okay, so your expectation or your hypothesis is that the two complexes will be identical that the complex described on GIS, or the idea of this immunoprecipitation is to identify a new protein in the complex. So what do you think are the chances to identify new
the
proteins?
idea is, well, in other organisms, EF6 is also present in other protein complexes, so we don't discard that in plants. And also, we know that it is impossible to find the same NUI4 complex in plants due to the fact that there is one protein that is not homologous in plant in that complex.
Okay.
So the idea is to try to elucidate if EF6 is also in other protein complex.
Okay, thanks.
Okay. We still have 5 minutes for questions, so if anyone, we're a little bit ahead of schedule. So if anyone wants, please ask. Otherwise, we can jump to the next. Okay. Okay, if anyone has any other question, then I think we can go ahead with the seminar. So thank you, Loretto. And
Thank you.
I will now introduce our next speaker, Alberto Cospinuela, who studied biotechnology at the Technical University of Madrid, and later did a master's on evolutionary biology at the Complutense University in 2016. And well, from 2015, he has been collaborating with Israel Pagan, and until he started his PhD with him in 2018. So thank you, Alberto, and over to you. We can't hear you. I don't know if you are muted.
Okay.
Yeah, perfect.
Okay, as I was saying, thanks everyone for attending to my presentation, and thanks Alvaro for introducing me. And okay, my talk is going to be about all the determinants of plant virus transmission. And first of all, I want to make clear the importance of plant virus infection. And the first thing is that plant viruses are important pathogens of crops. Infectious diseases cause approximately 16% of losses in crop production every year worldwide, and a third then are caused by virus infection. The virus infection does not usually come to the death of the plant, but instead in a diminish of production in fruits, seeds, and also the quality of the product. This is also a problem not only for crops, but also for wild ecosystems, as plant viruses reduces the fitness of their host, and every species of plant would respond differently from the virus infection. So the most susceptible ones could be reduced in number over time, and in some extreme scenarios, some species could be extinct in the wild due to the virus infection. So, as viruses, as parasites, viruses needs the ability to be transmitted, and this is a key component on its fitness. The way that viruses could achieve transmission are two. The first one is horizontal transmission, in which the virus is transmitted from host to host in two ways, either by contact or by vectors. And the second way of transmission is vertical transmission, in which the virus is transmitted from the parents to the offspring, in this case via the infected seeds. Most studies has focused on the determinants of horizontal transmission, and very little is known about the vertical transmission. And this makes a huge contrast with the problems that could be derived from vertical transmission, as the infected seeds provide the virus with a mean to persist long periods of time, and also to travel long distances due to, for example, the global trade of seeds. Also, infected seeds act as a source of primary inoculum, because even at low seed transmission rates, the plant virus could initiate damaging epidemics. All of this, along with the fact that more than 25% of plant viruses are transmitting through seeds, makes seed transmission a key component of virus epidemiology. And so, as I said, very little is known about the determinants, so we have chosen one plant, Arabidopsis thaliana, and two viruses, cucumber mosaic virus and turnip mosaic virus, in order to shed some light in this field. So my thesis objectives will be the identification of seed transmission genetic determinants, both from the side of the virus and the side of the plant, and later, as a secondary objective, I have to study the effect of co-infection on seed transmission, as this is a very frequent situation in crops and also wild systems. So the host will be Arabidopsis thaliana, that is a little annual plant with a short and well-characterized life cycle, and also a huge seed production, making it a perfect model to study seeds, and in our case, seed transmission. And its autogamous characteristics also joined with the huge genetic variability that it has due to its worldwide distribution, especially in the Northern Hemispherium, makes it an ideal organism to study plant genetics. In our case, the genetic interaction with the two viruses that we have chosen that are infecting it in wild ecosystems. So we have, in this case, more than 3,000 genotypes identified of Arabidopsis and more than 1,000 fully sequenced genomes that we are going to use later. In the case of the viruses, we have firstly turnip mosaic virus, or TBMV, that is a potivirus with a filamentous structure and a single-stranded positive sense RNA genome that is coded as a polyprotein that needs to be processed before expression. And this virus infects more or less 318 different plant species. So it has a quite wide range of hosts. And on the other hand, we have a Boomer mosaic virus, or CMV, that belongs to Bromobidirae family. It has an icosahedral particle, and once again, a single-stranded positive sense RNA genome, but this time it is segmented into 3 genome fragments that are individually encapsulated. And in this case, CMV is the plant virus with the widest host range, infecting over 1,200 plant species. So at first, these two viruses may seem very different, with different structures, genomes, and epidemiology, but they have a common point, that is one of their hosts, Arabidopsis thaliana, in which they both transmit both horizontally and vertically, and occurs in the wild and also in experimental conditions. So, in order to study further, that's the first objective. We have first talk about how viruses could achieve seed transmission. The most important thing is the virus reaching the reproductive organs of the plant, and this could be done in 2 ways. The first one is the direct seed invasion, in which the virus enters the embryo across the seed suspensor. And
let me, sorry to interrupt, Alberto, but the diapos are not passing. Oh. We have been for a very long time in the introduction to Arabidopsis and haven't seen the other diapositiva, so please take care of that.
Oh, sorry. Are you still seeing the same slide? Yeah, no?
Yes.
Yes.
Maybe I could try to share the screen again. Can you see it now?
No.
No.
No. Yesterday I have this problem, but I think that I solved it. But once
maybe
again,
you can get out and get in again.
Yeah, I will try. I will try that. Okay. Vamos a ver. Sorry about that. Vamos a ver. So eso, let me. Okay, let's let's try it again, I think. Yeah. Can you see it now?
Yes.
It's okay.
Okay, thank you, Alberto.
Okay, so the slides regarding the Arabidopsis themes are this one, and also the virus one, and this one that I think that it could help a lot seeing the structure and also the genomes. So maybe take a little time, certain needs. And if this happen again, please let me know. I hope not, but okay. Okay, let's continue with the ways of entering the seed. So as I was saying, the virus could reach the reproductive organs of the plant via direct seed invasion, infecting the embryo across the seed suspensor, or indirectly by entering the ovule or the pollen. These two factors, these two steps, needs to be done in time after before the suspensor starts its program cell death, and before the gametogenesis is completed and also the ovule and the pollen is released. And so this leads us with the theoretical seed transmission determinants that will be related at first with the virus capacity to reach reproductive organs in time as the virus speed of movement, the virus capacity to invade reproductive organs with traits such as the virus multiplication level, and later the traits related with survival of the infected seed embryo in order to allow the virus to remain in the seed while it's viable. So there are some theoretical seed transmission determinants, as I said, but there is a huge lack of experimental evidence. So in order to solve that, we have chosen 18 Arabidopsis genotypes, 2 isolates of TUMB, the UK1 and GPA1, and 3 isolates of CMB, the LS, the FNY, and the E72. And then we have measured the seed transmission in all of these Arabidopsis genotypes and in each virus isolate. Later, we chose 6 Arabidopsis genotypes out of the 18, phased them against one of the isolates of each virus, the GPN1 and the FNY. And then we have measured the seed transmission, as in the later experiment, but also the parameters related with kinetics of virus spread, viral accumulation, and effect of infection in rosette, inflorescence, and seed weight. So, having done this, we show that in the left we have the seed transmission of the 2 isolates of turnip-mosaic virus, and on the right the 3 isolates of CMV. And this data showed that seed transmission depended both on the virus species and also on the Arabidopsis genotype. So this suggests that there is a genetic effect between the virus isolate and species and also the Arabidopsis genotype. So, could be said that the virus seed transmission is modulated by a genetic factor that relies both in the virus and the plant. So to further explore this, we continue with results of the selection of 6 Arabidopsis accessions, in which we saw similar variation in the infection traits gathered as with the previous transmission values. And we then constructed a series of linear models taking into account all combinations with the infection traits that I described earlier. And then we perform a model selection in order to choose what model predicts better the seed transmission of each of the isolates selected. So first one, the model of GPN1 included the viral accumulation in the inflorescence and the speed of movement as their main predictors, as we can see here in the diagram of the relative importance of the factors, and the virulence of the virus as a third most important factor. And in the case of FNY-CMV, we have a similar case with the viral accumulation in the inflorescence and also the speed of movement being the most important factors, followed by the seed survival in this case. And then to go a little bit deeper in this, we have made some bivariate regressions in order to study the relationship with the most important variables and the seed transmission of these viruses. And in both cases of FNY-CMV and GPN1-GMV, we saw that the higher the speed of movement and also the multiplication in the inflorescence were, the higher the seed transmission was also. And the opposite occurred with both the virulence and the seed survival parameters, in which the less were the parameters, the higher was the seed transmission of both viruses. So with this data, it could be argued that our models, although predict well the seed transmission in these two isolates, could not be predicting the seed transmission well in the other isolates or in the other Arabidopsis genotypes. So we took the data from the 18 Arabidopsis genotypes that I showed before and simulate the data of seed transmission in these Arabidopsis genotypes and in the rest of the various isolates using the previous models that I showed, and then to establish if the prediction was well enough, we did a bivariate regression between the predicted seed transmission and the experimental one. And in both cases, we saw that the prediction was fairly well, as seen in the regressions here, meaning that these models are able to predict not only the isolates with which they are constructed, but also the other ones. And also meaning that the seed transmission determinants seen as important in the isolates GPN1 and FNY should also be important in the UK1, LS, and the 72. So they are a pretty good global models. So carrying on with the next objective, we are now focusing in the side of the plant. And in order to study the genetic determinants of seed transmission in the plant, we take the approach of a genome-wide association study, in which a genotype, in our case, the genotypes of Arabidopsis thaliana, and a phenotype, in our case, the seed transmission, was crossed in order to find relationships between the single nucleotide polymorphisms appearing in the genome of Arabidopsis and the natural variation gathered in the phenotype trait. So in this experiment, we choose again the FNY-CMV isolate and then 151 Arabidopsis genotypes, all coming from the Iberian Peninsula. And then we gathered all the infection traits as in other experiments, and also the seed transmission of this isolate in all of the Arabidopsis genotypes. So until now, we have the data of 62 Arabidopsis genotypes, seed transmission, and we ran an analysis, a Cuevas analysis, using the seed transmission firstly as a qualitative variable, so coded as the ability of being transmitted or not. In this case, 56 Arabidopsis genotypes showed transmission, so FNY transmission, and 6 didn't. A lot of significant SNPs, SNPs were found, but after filtering, looking for the gene function, we kept 7 genes associated with reproduction, stress response, defense, and also CMV-specific responses. Also, we conducted a second analysis, this time considering seed transmission as a quantitative variable, measured as the seeds infected, the percent of seeds infected by the virus, ranging from 5% to almost 0. Oh, sorry, 0 to 16%. And in this time, after all the filtering of the SNPs, we kept 3 genes associated again with reproduction, defense, and CMV-specific responses. These were the genes. In the side of seed transmission ability, we have Cal11 related with pollen co-development, and the other 6 genes were related all with defense responses. And AA50 with stress responses in general, Lecker71 with defense responses, Quirky 30 and FOD2, both related with salicylic acid mediated response against infection, and also specific responses against CMV, and finally the CL4 and AU10 related with response against CMV via gene silencing mechanisms. And using seed transmission efficiency, we have the A6 gene related with pollen co-development again, the 8-min7 related with salicylic acid again, and GL2, sorry, related with response against CMV and also salicylic acid response. So finally, could be said that we have 2 genes related positively, or at least theoretically positively, with vertical transmission, and these genes are related with the pollen development and viability. So the ability to the virus being stable in the pollen, and then we have the rest of the genes that theoretically could be related negatively with the vertical transmission, as they are all participating in defense responses and the control of the infection. So finally, we reach the third objective of the thesis, that is the study of mixed infection on effect on seed transmission, and this time we chose again the JPN1 UMB isolate and the FNY CMV isolate. Chose 6 Arabidopsis thaliana genotypes, and then in the experiment we have every single scenario of infection. We have the single infections as a starting point in order to compare them with the 2 types of mixed infection that are co-infection, in which both viruses enters the plant simultaneously, and superinfection, in which one virus, the second one, enters the plant once the first virus has established the infection in the plant. So the objective here is again measure the effect of the mixed infection due to in seed transmission. So we gather again the infection traits as in all of our previous experiments, and also measure the seed transmission efficiency. So this experiment, as well as the UAS, is yet to be completed. But this time in this experiment, we have gathered all the data, so only the analysis remained. Preliminary analysis show that there is an effect of co-infection and also superinfection on seed transmission efficiency. So things are singing well, I think. And finally, the conclusions of the talk would be that the efficiency of TUMB and CMV seed transmission, both dependent of the Arabidopsis thaliana genotypes and also the virus species interaction, the virus speed of within host movement and its level of multiplication in inflorescence were the best estimators both of seed transmission, both in TUMB and also CMV. And finally, plant genes related to chloroplast signaling, silencing, pollen formation, and salicylic acid mediated responses are associated with FNY-CMV, seed transmission efficiency. So, thanks everyone in the lab 227 for the support, and especially to my director, Israel, and my technicians, Marisa and Miriam, because without them, none of this work would have been possible. So thank you, everyone. And finally, thank you all for your attention. And if you have any questions, I will be delighted to answer them.
Thank you, Alberto. We have 10 minutes for questions, so go ahead.
Alberto, I have a couple of questions. The first one is, since clearly the level of virus in the inflorescence is critical for seed transmission in both type of virus that you have studied. So do you guess that maybe just to have some modification of cell wall of cell-to-cell transmission and vascular or alteration of the vascular system will reduce or will have some impact of the reduction of the level of the virus in the inflorescence, or are you think about some developmental program that can reduce this?
I think that maybe the modification of vascular system could have more of an impact in the speed of movement more than in the multiplication, because both viruses move through phloem, so it needs to interact with the plasmodesms. So maybe the most affected factor due to this modification could be the speed of movement, also an important factor, but I think that the multiplication and accumulation of the virus is controlled more by the defense responses more than the proper configuration of the organs of the plant, I think.
Okay, so the reason I was asking this is because some of the genes that you have classified as salicylic acid related could be also directly or indirectly related with cell wall modifications. That's one of the reasons I'm asking.
The salicylic acid ones, there are a few publications that has found a relationship between the salicylic acid presence and also the limitation of the viral movement. So once again, I think that the salicylic acid ones would be reducing the movement of the virus. Okay. So directly affecting the reach of the reproductive tissue, but more due to the movement and not to the multiplication.
Okay, thanks.
At first.
Fernando raised hand, so please, Fernando.
Yes, I have a couple of questions, or even three, if I may. One is about this last thing that you were discussing with Antonio Molina, and that seems to be an important factor in seed transmission, that is speed of movement. Mm-hmm. But I'm not so familiar with this concept. So what do you call exactly speed of movement?
So
the time the virus getting systemic, the time the virus gets to the flower, what do you call speed of movement?
I think that it's easier to explain how it was calculated.
Okay.
Yeah, we took the plant in several points of development and then fragmented and measured the presence of the virus in each of the fragments. So when the plants, I think that was every 2 days, 5 plants were selected and then fragmented into 3 pieces. And then the presence of the virus was detected in one of each these pieces. And this process was repeated over 30 days. So we have an idea of where the virus is in the plant in several stages of the development.
Okay.
So it's, the measure is in centimeters of the plant per day is the units.
Okay. Okay. So the plants, but the plants will not be the same size or height because they are from different genotypes, right?
No, and that's because we fragmented it into 3 equal parts and not selected one measure like 3 centimeters, because maybe 3 centimeters is a lot for one accession that takes long to develop and a little from one that is already big. So we did it proportionally to the size of the plant.
Okay, and the second question is related to this identification of 2 genes that are related to pollen formation and viability. And your slide about the possibility of virus entering the seed. So do you think that this suggests that the pollen has a role in the access of the virus to the seed?
With these results, suggests that the pollen has a role, but not which role it has, because the genes are related with the pollen formation, especially with the coat of the pollen. But we don't know if this facilitates the entering of the virus or the opposite. So there is an effect, but we don't know in which direction is the effect.
Yeah, I understand, because the 2 factors that you identify, both the speed of colonization or of movement, or the virus accumulation in the flower, would suggest that the entrance is through the mother sporophyte, right?
We haven't tested with such precision, so we don't know. We don't know if it's preferably via embryo or via pollen or ovules.
Okay.
CMV could achieve the 2 ways, so it's not as exclusive, I think. But I don't know which way is the predominant in this case.
Okay, so because you don't know if these 2 viruses infect the embryo.
Yeah, CMV infects both the embryo and the pollen and the ovule. But with TUMB, we don't know. We don't know because there is no previous description of TUMB seed transmission, either in Arabidopsis or other hosts. So there is very, very little known about the TUMB seed transmission.
Okay, thank you.
Thank you.
Okay. I think we have time for one more question before we go to the next talk. Otherwise, we can just move ahead.
Okay.
Okay, cool. Bye.
So thank you, Alberto.
Thank you all.
Okay, so our next speaker is Laura Bouza Morcillo. She studied biology at the Complutense University. Afterwards, she did her master in agroforestal biotechnology at the UPM University and her master's thesis with Luis Oñate. Then she spent 2 years in the Institute of Agrobiological Investigation of the CSIC in Santiago de Compostela, and she is currently doing her PhD thesis at José Jarillo and Manuel Piñeiro's lab with an FPI grant. So Laura, if you want to start.
Yes, hi. Sorry, I have problems with...
Don't worry, you have time.
Okay.
Okay, perfect.
So good morning, everyone. Thank you, Clara, for your introduction. As she has said, I'm doing my PhD at José Antonio Jarillo and Manuel Piñeiro's lab. And I'm going to talk about the role of the protein enhancer of polycon-like in the epigenetic regulation of two developmental processes in plants. The research in our lab is focused on elucidating the molecular basis regulating developmental phase transitions in plants. In this context, we are especially interested in the epigenetic regulation. This regulation is mediated by chromatin remodeling complex processes that are carried out by complexes which are able to modify the chromatin state by different mechanisms such as histone acetylation or histone exchange of canonical histone by histone variants. This epigenetic regulation plays a fundamental role in establish and maintain gene expression patterns that control developmental and biological processes such as the regulation of flowering time. As an example of how a chromatin modification causes a change in the transcriptional state of a gene related with flowering, a protein called early ensordase is the one that belongs to the chromatin remodeling complex Swar1, substitutes the canonical histone H2A by a variant called H2A-SID. The deposition of this histone on the FLC chromatin is required for the expression of this floral repressor. In fact, the loss of SD1 function causes an early flowering phenotype in the mutants. This process is a complex regulatory mechanism that has been associated to histone acetylation by another putative acetyltransferase complex that shares some of the subunits with Swar1 and has a functional relationship with it, which is called NUI4. And as my partner Loretto has mentioned before, this histone acetyltransferase complex has been described in yeast and is the responsible of the acetylation of histones H4, H2A, and H2A-SID. Most of the subunits of this complex are conserved in Arabidopsis thaliana, and I'm particularly interested in those that are part of the piccolo NUI4 subcomplex, which contains the catalytic subunit ESA1 in yeast and the catalytic subunit in plants that seems to be ham. Little is known about the function of the conserved subunits of the complex in plants, and I have focused my thesis in the characterization of one of them, that is an enhancer of polycon-like 1, EPL1. We chose this protein because it's exclusive of the NUI4 complex, and in yeast it has essential functions providing a scaffold to the piccolo module and guiding the catalytic subunit ESA1 to the chromatin. This protein is conserved across in plants and humans, where it belongs to the TIP60 acetyltransferase complex. Interestingly, it presents an N-terminal domain, which is conserved in all the species, and is the responsible of the union with the catalytic subunit and to the nucleosomes. One curious data of this protein is that in humans it's related to cancer as its interaction with the transcription factor E2F1 promotes the proliferation of tumoral cells. EPL1 in yeast is highly conserved for the species, and most plant genomes include genes with high homology to the yeast NUI4 genes. In Arabidopsis thaliana, there are two EPL1 proteins that we named EPL1A and EPL1B, and present a high level of homology, nearly to the 75%. As I mentioned before, I have focused my thesis in the characterization of this EPL1 protein in Arabidopsis thaliana, and in this context, we have defined three main objectives. The first one was defining the composition of the EPL1 containing chromatin remodeling complex that regulate flowering time in Arabidopsis. The second one is the function and characterization of these proteins. And related with these objectives, we have defined two different lines of investigation. One of them related with the role of EPLs in the chloroplast biogenesis, and the other related with the role of these proteins in flowering time regulation. And also the third objective was the genetic dissection of the EPL gene functions in the regulation in flowering time and in other developmental processes. First, and related with objective 1, we started our work demonstrating that plant EPL1 proteins may serve as a scaffold for piccolo proteins. For that, we performed monoprecipitation experiments in tobacco, and apart from the interaction between EX6 and EPL1A and EPL1B and EX6 with ING2 that Loretto showed you before, we have also identified the physical interaction between both EPL1 proteins and two other putative homologues in plants, which are HAM1, that is the catalytic subunit, and ING2. So these results put in evidence the possible existence of a uniform complex in plants, or at least the piccolo submodel. Next, we established knocked out the mutant lines on the expression of either EPL1A or EPL1B and characterized them in some grain conditions related with different photoperiods or temperature. As you can see, at normal temperature, there's a slight, no significant delay in flowering in EPL1B mutants at London conditions, but there is no phenotypic alterations in single mutants at short-day conditions. However, when plants are grown at low temperature conditions, the late flowering phenotype observed in EPL1B single mutants is much more obvious and significant, while there is no phenotypic alterations in EPL1A mutants at these conditions. Also, and regarding the fact that EPLs have a high level of homology, but they don't have exactly the same phenotype in flowering, we thought that they could be partially redundant. For this reason, we decided to generate double mutant lines for these genes. The double mutant lacking the expression of both EPL1 genes shows severe phenotypic alterations. As you can see, the plant has a lot of problems to growth. It has albino seedlings of reduced plant size. It has defects in leaf development, chlorosis, and light hypersensitivity. And in addition, these plants are not able to produce viable seeds, something that forces us to work with sesqui mutant populations that produce only between 5 to 8% of these double homozygous plants. So these results indicate that the EPL1 homologues are redundant. And another evidence that supports this idea is that we are able to complement the phenotype of the double mutant when we generate transgenic lines expressing target versions of in single target versions of one of the EPLs under the control of their own promoters. So these two phenotypes observed in the singles and double mutant plants open to us two different lines of investigation. We have focused previously our attention in the characterization of the double mutant. First, we characterize a bit further the developmental defects caused by the loss of both the EPL1 function, and we observe a significant reduction in chlorophyll content in the leaves of mutant plants. And indeed, in confocal microscopy analysis, we show that although these mutants have more or less the same number of chloroplasts, the plastids seem to be smaller and display the reduced autofluorescence. And a closer look into the plastid ultrastructure using transmission electron microscopy confirms the abnormal development in the mutant, with plastid of a smaller size and disorganization or problems to form thylakoid and grana. So chloroplasts develop from etioplasts in response to a light stimulus as part of a developmental program called photomorphogenesis. And we decided to study the regulation of EPL1 expression during this process. For this, we grew up plants during 5 days in darkness, and then we picked up material after different hours of light exposition. As you can see, also the transcript levels of EPL genes did not change after illumination with light, the proteins levels of EPL1B change significantly. So in higher levels at the dark to light transition and then progressively decaying, peaking out after 24 hours, and then the protein levels recover after 2 to 3 days. This post-translational regulation suggested that NOE4 activity may have a role in the early stages of chloroplast biogenesis. So to understand why chloroplast development was altered in the EPL1 double mutant, we performed a transcriptomic analysis. We identified more than 5,000 differentially expressed genes, and as an interesting observation, RNA samples from the mutants seem to lack plastic ribosomal RNAs, some of which are transcribed by plastic RNA polymerase. Also, among the genes that were downregulated in the mutant, we observed an enrichment in GO10s associated with chloroplast function. Indeed, the level of expression of total plastic genes encoded by the nuclear genome compared to a wild type was significantly reduced. This data suggested that it could be a transcriptional deficiency of plastic genes in the double mutant. So, as plastic transcription was one of the enriched annotations in our transcriptomic analysis, the first thing we hypothesized is that the plastic RNA polymerase activity might be inhibited. The control of chloroplast development is mediated by 2 RNA polymerases. 3 of them are encoded by the nuclear genome, for example, SK3, and the rest are encoded by the plastic genome. There are also cofactors of these plastic RNA polymerases that are encoded by nuclear genome, for example, Mrg proteins. Transcripts of these proteins are translated in the cytoplasm and enter to the chloroplast, and there nuclear encoded RNA polymerases promote the expression of plastic encoded RNA polymerases, and this together with cofactors coming from the nuclear genome from a functional plastic RNA polymerase that transcribes the rest of chloroplastic genome. So first we checked in our transcriptomic analysis genes related with plastic transcription that were downregulated in the EPL1 double mutant. We identified the SK3 that encodes one of the 3 nuclear RNA polymerases and a number of cofactors related with essential for the activity of the RNA polymerase encoded in the plastic genome. In addition, SK3 mutants and some of the mutants lacking of these cofactors have a phenotype very similar to our double mutant. So we reasoned that maybe plastic RNA polymerase activity might be inhibited in the EPL1 mutant, double mutant. So as this activity seems to be reduced and in double mutants, and since EPL1 is the functional scaffold for the picoron module, which fetches the unacetylase activity of the NOE4 complex, it was reasonable to hypothesize that the loss of EPL1 function might bring a decrease in the histone acetylation of our target genes. To check this, we performed chromatin immunoprecipitation experiments that briefly consist on analyzed by qPCR the enrichment in DNA of immunoprecipitated fraction of chromatin using a specific antibody against a histone mark or a protein in particular. In this case, we use an antibody against the acetylation of lysine 5 on histone H4, which is a chromatin modification characteristic of the NOE4 complex. And we analyze different regions relevant to nucleus and plastic RNA polymerase encoded activity. In this case, we analyze SK3, PTAC12, and PTAC14. And we identified lower signal in EPL1 dimutant samples for several regions at this loci. So we can conclude that EPL1 regulates acetylation of H4 at this gene. Next, we want to test if these genes were direct targets of EPL1 proteins. For that, we have generated Arabidopsis transgenic lines expressing target version of the EPL1B protein under the control of a constitutive promoter. And we perform these tests using an anti-HA antibody, and we observe the binding of EPL1 proteins to this loci. So if this is consistent with our hypothesis that the Mrg complex directly regulates the expression of these genes. So finally, we propose a model in which the shift from heterotrophic to autotrophic growth requires a modification of chromatin as a specific loci, which are essential for the activity of the plastic RNA polymerases. Histone H4 acetylation at this loci enables plastic transcription and chloroplasmagenesis. So EPL1 and possible the Mrg complex plays a major role in the coordination of nuclear and plastic genes, plastic genomes, and without this activity, the plant is not able to complete the life cycles. The study of the role of EPL1 in chloroplasmagenesis have been one of my of the main objectives of my thesis. But at this moment, I'm focusing my attention in elucidating the possible function of this protein in a control of flowering time. As I mentioned, I showed you before, EPL1B mutants have a late flowering phenotype at low temperatures. To understand why flowering time was altered at a low temperature in these mutants, we performed a transcriptomic analysis in which we found 400 genes, upregulated genes, and 53 downregulated genes. We focus our attention in this last one category because we thought that the late flowering phenotype of observed could be due to a loss of the acetyltransferase activity of mutant in these mutants. So in this category, we found only 2 genes related with flowering time regulation, which were FT and Barrero FT. We checked by qPCR FT and other genes related with flowering time regulation, such as constans, SOC1, or FLM. And we found that FT is significantly less expressed in the EPL1B mutant. This gene is one of the most important floral integrators. It's part of the phlogiston and acts as a switch for flowering initiation. Indeed, at low temperatures, while the plants present a delay in flowering, a phenotype that has been directly related with reduced levels of FT expression. In fact, FT mutants display a very late flowering time phenotype at low temperature. And we saw that maybe a molecular relation between FT and EPL1B might explain the late flowering time phenotype that we observe in the mutants. The regulation of FT expression at low temperatures is mediated through different pathways. And as a brief explanation, COG1, that is a ubiquitin ligase, degrades gigantea, which is a FT activator. And moreover, expression of SBP and FLM in response to low temperature repress FT expression. As an example, SBP modulates FT expression by direct binding with each promoter. Then, in an opposite way to EPL1B, SBP mutants show early flowering time phenotype at low temperature. We are now performing genetic approaches in order to unveil a possible relationship between EPL1B and some of these flowering regulators. For that, we are generating crosses between knockout mutants of these genes and the EPL1B mutant. EPL1B and SVP genetic interaction analysis is more advanced, and we have demonstrated that SVP genetically interacts with EPL1B during flowering, since the double mutants have an early flowering time phenotype. This demonstrates that SVP and EPL1B are in the same molecular pathway, and that SVP is downstream of EPL1B. Moreover, and regarding the possible relation between EPL1B and FT expression, we have done a preliminary chip against a histone mark in order to determine if lower transcription levels of FT found in a mutant are related to less acetylation levels in this locus. As very previous results, we have observed a lower signal in some region of this gene. And however, these are, as I have said, preliminary results, and we are working now in repeating these experiments. So finally, and as a brief summary, we have demonstrated an essential function of EPL1 genes in the regulation of chloroplast biogenesis by controlling the acetylation levels of key genes involved in plastic transcription. And in this part, in the future, we will perform genetic analysis of the interaction of EPL1 genes with these targets that we have identified mediating chloroplast biogenesis. And also, we have identified a role of EPL1B gene in the promotion of flowering time at low temperature. We have a lot of work to do in this part, but we will more or less, we will carry out the genetic analysis of EPL1B cross with other mutants affected in flowering time response. We will determine the interaction network of EPL1B with other regulators of interest. We will corroborate the altered acetylation levels of FT promoted in EPL1B mutants, and also if there is the direct binding of EPL1B to FT chromatin, and reveal the EPL1B binding sites at genome-wide level. So finally, I would like to thank all the members of my group, and especially mention to my supervisors, and a very special mention to Javier Barrero, since we have developed all this work together. So I will be happy to take your questions.
Okay, thank you, Laura. If is there any questions for Laura, just go ahead and unmute your audio. Okay, Fernando, do you have a question for Laura?
Yes, thank you. Laura, I have a question, because it seems that both the component that you analyze of this Piccolo complex and EAF6 of Loretto affect flowering and affect chloroplast biogenesis. So the question is there any link between chloroplast biogenesis and flowering, or is it rather that these proteins have such a basic role that they would affect many different things, and you are just focusing on these two traits? So
I think it's more the second one. Well, at least APLs it seems to be affecting a very basic development moment of the plant. So maybe this in the rest of the development of the plant, they affect other mechanisms, for example, flowering. But I don't know if EAF6 has the same relevance in the chloroplast biogenesis. Maybe it would be interesting to check in the future.
Well, there was a slide of Loretto in which it said it affected also chloroplast biogenesis. This is why I thought if it was a possible link or not.
Yeah,
but
I
the thing
think
is rather that the effect is very basic, and then there are many different things that are affected. Okay,
yeah.
thank you, very clear.
Okay. Is there any more questions for Laura? Okay, we have a few minutes, so if anybody wants to ask anything else, we have 5 minutes left. Okay, so if there is nobody else, thank you, Laura, for your presentation. And we are gonna go ahead and continue with our session. Okay, so our next speaker is Javier Barrero Gil. He did his PhD research project in the UPM under the supervision of Professor Alonso Rodríguez Navarro and Begoña Benito. His research was focused on ion homeostasis in lower eukaryotes. Then he moved to Arizona to study sodium toxicity in plants at the lab of Professor Karen Schumacher. And after 2 years, he returned to Madrid to work with Professor Julio Salinas in plant cold stress responses at the CIB. And he's currently working at Manuel Piñeiro and José Jarillo lab, focusing on the molecular mechanisms underpinning developmental phase transitions. So Javier, whenever you want to start.
Okay, can you hear me?
Yes.
Okay, let me share. Compartir.
Okay.
Okay. Oh, sorry. All right, here it is. So first, I would like to start by thanking Elena and the organizing committee for inviting me to present some of the work that I have been carrying on at CBGP. And today I will present one of the work that I have performed in one of the lines in these 3 years that I have been working at CBGP. So in a moment of particular inspiration, I entitled my talk, "The Road Not Taken, a chromatin switch chooses between stress tolerance and reproduction." And I'm afraid I have to stand by the title and explain why it is so. So the title refers to a poem by Robert Frost. It's called "The Road Not Taken," which ends with the stanza, "Two roads diverge in a wood, and I took the one less traveled by." And that has made all the difference. And I started with this because I think it illustrates very well the dilemma that monocarpic plants face. So let me explain why. The idea is the poem has some imagery referring to diverging paths, also about the difficulty of foreseeing the merits of each path, and also about the consequence that choosing one path over the other it's going to bring. And I think this is very relevant to plants in terms of especially monocarpic plants that flower just one time, in terms of choosing between remaining in a vegetative stage or committing to reproduction, because choosing between one path or the other is going to have dire consequence in terms of plant survival and also in terms of reproductive stress, especially when the resources are limited or when the environmental conditions are not optimal. And today I'll be presenting a chromatin switch that we propose is at the center of this decision. So by now you know that in the lab we are interested in developmental phase transitions, and so plants during their life cycle they go through different stages that are have particular morphological and particular morphologies and particular physiologies, and transition from one stage to the next, it's a process that is tightly regulated, and also it's informed by endogenous and environmental cues. Typically, these transitions involve large changes in gene expression patterns, and research in the past decade has demonstrated that epigenetic regulation and chromatin remodeling complexes play an important role in these transitions. So earlier today, we had the opportunity to learn about the work of Loretto and Laura, that by now we are familiar with the way these chromatin remodeling complexes can shape gene expression patterns. So for example, Laura showed us that that histone acetylation is critical in the transition from dark to light in order to coordinate nuclear transcription and plastic transcription. On the other hand, Loretto also showed us that histone acetylation activity is important to promote flowering under short days. So it becomes clear that histone acetylation regulates plant developmental phase transitions. But the thing is that histone acetylation is not the only epigenetic mark that is important for those kind of regulations. So in fact, there are multiple post-translational modifications of histones, including acetylation, methylation, phosphorylation, and ubiquitination. These post-translational modifications takes place in multiple residues, namely lysines, arginines, serines, threonines. They are reversible and they are highly dynamic. So these post-translational modifications are highly dynamic, and and the combination of all these marks sometimes are called epigenetic landscape, and the combination of all these marks is expected to regulate chromatin accessibility and structure. There's no doubt that if we were able to interpret and modify this epigenetic landscape, we would have a very powerful biotechnological tool for controlling gene expression. In this context, it is important to recognize, sorry for that, didn't mean that. Okay. It's important to recognize three classes of histone interacting proteins: writers, erasers, and readers. Writers are enzymes that catalyze the addition of a chemical group to histones. Erasers are enzymes that catalyze the removal of that group to histones. And also readers are proteins that feature particular domains that recognize and bind specific post-translational modifications. So Loretto and Laura have already presented the new A4 complex, which has features histone acetylation activity, and this complex contains several proteins featuring domains that recognize post-translational modifications on histones, like acetylation and methylation. And today I'll be focusing on the Mrg module that recognizes this epigenetic mark. So Mrg proteins were first characterized in yeast and animals, where it was they were reported to bind trimethylated lysine 36 on histone H3. This epigenetic mark is enriched. It's a characteristic of actively transcribed genes, and it's enriched over the gene body of these genes. And it has been related to transcription elongation, alternative splicing, and also preventing spurious transcription. In contrast, in plants, this epigenetic mark is more enriched towards the 5' UTR. And that suggests maybe a role in transcription initiation, although other roles of this mark cannot be ruled out. So in the lab, we started to work on Mrg's proteins a while ago. as part of the research project, PhD research project by Alfonso Mouriz. So Alfonso identified two Mrg homologs in Arabidopsis, named Mrg1 and Mrg2. And he identified knockout mutants in Arabidopsis, tDNA insertional mutants, and started to characterize the function by studying these mutants. The first thing he observed was that there were no obvious morphological alterations in single mutants, but there was a clear late flowering phenotype in the double mutant, Mrg1, Mrg2. This late flowering phenotype was characteristic of long day conditions. And when he started to analyze the expression of key flowering genes, he observed that he detected lower transcript levels for two important flowering regulators. These are FT and SOC1. So this observation is what's consistent with the phenotype. Now, because MRGs are readers of H3K36 trimethylation, it was reasonable to wonder if this late flowering phenotype was dependent on this mark. So to do this, what he did was crossing the double MRG mutant to SDG8, to a mutant knockout in SDG8. SDG8 is one of the main important, is the most important methylases that write this mark, that methylate H3K36. So he did the cross, he performed the cross and analyzed flowering in these mutants. And the results show that the SDG8 mutation is epistatic to MRG mutations, indicating that MRG function needed or required SDG8, a functional copy of SDG8 in order to show up. So he concluded that MRG role in flowering requires the trimethylation of H3K36. Now, in 2014, two papers came along, were published that describe MRG role in flowering. So the first one described MRG binding to the epigenetic mark in plants, for the first time in plants, to the trimethylation of H3K36, and describe also that upon this binding, the transcription factor constans, which is an important transcription factor for photoperiod-induced flowering, was recruited to FT chromatin. And the second report confirmed binding of MRG to FT chromatin via the mark, via H3K36 trimethylated, and also MRG recruited new A4 complex in order to acetylate histones at FT chromatin locus and to promote FT expression under long days. So these two papers provided a nice molecular explanation of the role of MRG on flowering. However, in the meantime, Alfonso had made an interesting observation, and that was that while analyzing transcriptomic data for the MRG double mutant, he realized that among upregulated genes in this transcriptomic data, there was an enrichment of genes related to biotic stress responses, namely cold, drought, and abscisic acid. And at that point, José Antonio Jarillo approached Julio Salinas, and I was working at his lab at that moment, and we started to collaborate in the project. So one of the things that interested me most at the time was that the CBF's transcription factors, which are one of the most important mediators of cold response in Arabidopsis, were upregulated in the MRG double mutant. So we confirmed that by studying CBF expression in the double mutant in control conditions, which was Alfonso's observation, and also in response to cold, to low temperature. So we confirmed that all 3 CBFs were upregulated in control conditions, and we observed that cold response was similar to the wild type. So it appeared to be specific of control conditions. In addition, not only the CBFs were upregulated in the double mutant, Mrg double mutant in control conditions, but also a number of genes that were reported to be regulated by CBFs. In fact, there was an enrichment of what we call the CBF regulon, that is the gene set of genes that are regulated by CBFs. So why all the fuss about the CBFs? Well, the CBFs are one of the major pathways controlling the ability of Arabidopsis to withstand freezing temperatures. So Arabidopsis is a plant from a temperate climate, and which are characterized by harsh winters, as we know. And as such, Arabidopsis can develop a process called coacclimation, which is an adaptive process upon which, or by means of which, upon exposure to particular environmental cues involving low temperatures and also some light, particular light conditions, the plants increase their tolerance to freezing temperatures, and they can withstand harsher conditions. In contrast, tropical plants like tomato, for example, lack this adaptive mechanism and are sensitive to freezing temperatures. Now, the observation that a plant, in this case the Mrg double mutant, has a higher transcript level of CBFs in control conditions, in non-stressful conditions, indicated or suggested that if we were to expose plants to freezing temperatures without giving them the opportunity for cold acclimation, probably the Mrg double mutant would have a higher tolerance to freezing temperatures than the wild type. That's called basal or constitutive freezing tolerance. And that's the freezing tolerance that display non-acclimated plants, okay? So we did this with the double mutant and the wild type, and as we predicted, the Mrg double mutant displayed a higher basal freezing tolerance than wild type plants. So we concluded that Mrg proteins negatively regulate basal freezing tolerance, probably because they had higher CBF transcript levels in non-stressful conditions. And the question was why the CBFs are up, and we immediately linked this to the fact, to the observation that Alfonso had made that SOC1 transcript levels were low in the double mutant, and with this we make that connection because it was years ago it was reported that the transcription factor SOC1 can bind to CBF chromatin and repress their expression. And indeed, there are studies, genome-wide studies of SOC1 and direct targets of SOC1. There is a collection of genes that are putative targets of SOC1. So we try to identify them on the transgenic, on the transcriptomic analysis, and we identify that not only CBFs were up in the double mutant, but there were also other 15 putative direct targets of SOC1 that were upregulated in Mrg, and this is enriched. So it was reasonable to predict that there was some kind of functional link between SOC1 and CBFs. SOC1 and CBFs, sorry, between Mrg and SOC1. And to check this, we did a genetic interaction experiment. What we did was crossing these mutations. We crossed SOC1 with the double mutant Mrg1 and Mrg2, and we started by analyzing the expression of abiotic stress response genes in these mutants. the results show that all 3 mutants have a similar gene expression patterns of abiotic stress response. So all of them are induced related to the wild type compared to the wild type plants. And also the combination of SOC1 and Mrg mutations do not show any clear additive effect. So this supports a link between SOC1 and Mrg genes. However, to demonstrate the link, we had to compare basal freezing tolerance. And this is the experiment that I'm showing here. And the results show that the increased basal freezing tolerance that we observe in the double mutant required an active copy of SOC1. And if you don't have it, the freezing tolerance is decreased. So the conclusion is that MRGs negatively regulate constitutive freezing tolerance or basal freezing tolerance in a SOC1 dependent manner. So because MRGs are histone readers of the H3K36 trimethylated mark, we reasoned, and because there was a regulation, a functional link between MRGs and SOC1, we reasoned that maybe MRGs were binding to SOC1 chromatin to somehow regulate its expression. And to check this, what we did was a chromatin immunoprecipitation experiment using a transgenic line expressing a target version of MRG2 fused to the YFP. And we use an antibody against GFP to immunoprecipitate chromatin. And by PCR, we would check the presence in the immunoprecipitated fraction of different regions of the SOC1 chromatin. We did this experiment, and the results show an enrichment of a particular region of the SOC1 chromatin in the transgenic lines, indicating that Mrg proteins, at least Mrg2, is able to bind to SOC1 chromatin. Now, because MRG proteins bind to SOC1. So the question now is, how do they regulate its expression? And because MRG proteins have been reported to associate with the new A4 complex, we could also think or consider that maybe MRGs were regulating the acetylation status of SOC1 chromatin. And we check this by another chromatin immunoprecipitation experiment, but this time using an antibody raised against the polyacetylation of diverlysins on histone 4. And once again, by PCR, then again, by PCR, we show that there was a higher recovery in Columbia plants compared to the mutants of regions, of particular regions of the SOC1 locus, indicating that there were lower H4 acetylation levels at SOC1 in the mutant compared to the wild type. So by then we had established a functional link between Mrg proteins and SOC1, and also proposed a model, a mechanism that how this was happening. However, and all of this was in the context of freezing tolerance. However, during the course of our investigation, we started to realize that probably the SOC1 role in the abiotic response was wider. It was probably not restricted to freezing tolerance. And this notion was based on the analysis of genome-wide studies of SOC1, of both SOC1 binding and also of expression. So for example, in this study, it was described that among the genes that were putative targets of SOC1, there was an enrichment of genes involved in abiotic stress responses. And those genes were not particularly related to cold stress, but also with salt stress, drought, etc. And the other observation was that if we took that gene set of SOC1 putative SOC1 abiotic stress targets, direct targets, we observed that most of them were downregulated by SOC1. So it was a negative regulator of those stress. It appeared to be a negative regulator of those genes. So we decided that it was worth to test if the Mrg SOC1 module, regulatory module, was also regulating other abiotic stresses. And we did that, and we started with drought, and we observed that just by withholding water, that the double Mrg double mutant was displayed a higher tolerance to drought conditions than the wild type. And interestingly, SOC1, the SOC1 single mutant and the triple mutant also showed some degree of higher tolerance than the wild type. But when we compare the double with the triple, we observe that the increased tolerance of the double requires a functional copy of SOC1. So again, it seemed that Mrg double mutant, Mrg proteins were negatively regulating drought stress response in a SOC1 dependent manner. Then we wonder also about ABA response, and we perform a similar experiment, this time exposing young seedlings to ABA. And in the double mutant, we also observe a hypersensitive response to ABA compared to the wild type. And that hypersensitive response was dependent, again, of the presence of a functional copy of SOC1. The conclusion was that the Mrg SOC1 module negatively regulates abiotic stress responses, and probably, in general, probably by modulating either ABA signaling or ABA levels. Now, in order to provide some physiological context to these observations, we started to read in the scientific literature and realized that most of previous reports dealt with the way abiotic stress responses affected flowering, affected different developmental phase transitions, but in particular flowering. So, for example, it was reported that a short intermittent cold exposure delayed flowering. And on the other hand, moderate drought conditions accelerated flowering in a process it's well known, it's called drought escape. However, there were some models published trying to explain these observations, and it was interesting, this one related to cold response, in which it was proposed that on early spring or fall, where there are cold temperatures, CBFs are activated, and CBFs were demonstrated to activate FLC expression, which is a well-known floral repressor. And FLC inhibited SOC1, and so flowering was delayed and cold resistant was promoted. Later in the season, warm temperatures shut down CBF expression, and photoperiod and endogenous signals shut down FLC. So that releases SOC1 inhibition, and flowering is promoted. And also, in this case, SOC1 inhibition of CBFs was explained as a mechanism to ensure that any late, so that the flowering was not inhibited in case of any late frost event. However, we had discovered that SOC1 was involved role in abiotic stress tolerance was wider than initially thought. So we thought that required a revision of this model. And when we considered this, we thought, well, SOC1 is generally regarded as a mediator of a developmental phase transition, in this case, floral transition. So maybe it's that floral transition attenuates abiotic stress responses and not the other way around, which was the notion that was present in the literature. So we decided that we needed to put the Mrg SOC1 regulatory module in a developmental context. And in order to do that, we defined 2 stages: before floral transition and after floral transition. And in a series of preliminary experiments, we set 8-day seedlings as before floral transition and 12-day seedlings as after floral transition. And that was based on the expression of floral identity genes, or meiotic identity genes. And what we observed was that SOC1 differences, SOC1 expression, there were negligible differences in SOC1 expression in the Mrg double mutant compared to the Columbia before floral transition, but there were significant difference after floral transition. And also it was nice to check, to find that these different gene expression pattern of SOC1 in the Mrg mutant compared to the wild type also related to differences in H4 acetylation status. So we identify no difference in H4 acetylation status at SOC1 level between Columbia and the double mutant at 8 days, but there were clear differences in acetylation, in H4 acetylation status after floral transition. So with this in mind, we can summarize, and we can see that SOC1 has a double role in plant development, promoting floral transition, and also attenuating abiotic stress response. And what we propose is that the developmental switch that we have uncovered, operated by Mrg proteins on SOC1 locus, what does is that it promotes reproductive growth at the expense of costly efforts to adapt to challenging conditions. So we think that this switch is going to promote reproductive success at the cost of being less able to tolerate stress responses, stressful conditions. And with that, I leave you with that. But before finishing, I want to thank, I want to highlight the contributions of the people that have worked in this project. First, Alfonso Mouriz, that started the project. He developed all the transgenic lines and mutant lines, performed the crosses, and did all the analysis regarding flowering phenotypes. Raquel Piqueras helped along in some experiments in the flowering phenotypes and also in the part of abiotic stress response. And also to my supervisors, Jarillo, Piñeiro, and also Julio Solinas, that supervised the whole project and provided financial support. And financial support is acknowledged by the Ministerio de Ciencia y la Innovación. I'm happy to take your questions.
Okay, thank you, Javier. If there is any questions for Javier, please go ahead and unmute your microphones.
Okay, I have a couple of questions.
Mm-hmm.
The first one is regarding the fact that you say that there are some plants that are cold tolerant, while that they can trigger like this adaptation to cold, while there are others that can. So have you analyzed if those plants that cannot make this change, this adaptation, are defective in these Mrg genes, or do you think it's related with something that is more complex?
Well, I don't think it's that's a very good question. I think there's this regulation, this Mrg SOC1 regulatory module. It's general for all abiotic stress responses. So, for example, if we take a tomato, probably this regulatory module is not going to take effect because CBF, in this case, that are the effectors of this cold acclimation process, is not working in tomato. But probably if instead of analyzing freezing tolerance, we analyze, for example, salt tolerance, maybe we could see an effect. So it depends on, in each species is going to have a different abiotic stress responses. And I don't know, but I can speculate, and I would speculate that probably if we are right, if our proposal is correct, maybe there are other stresses that, and the Mrg SOC1 regulatory module would be important for them.
Okay, thank you. And I also have the second question regarding the phenotypes of some of the defective mutants for Mrg. And the thing is, like, the phenotypes look quite normal in the compared with. Have you analyzed if there is like any trade-off on the stress response triggered by these genes, or is something that we like already know for sure, or there is not?
We haven't analyzed a trade-off because that's a complicated experiment and we haven't performed it. But having said this, it has been reported that there is a trade-off, published trade-off of SOC1 not SOC1, about a trade-off between being able to tolerate drought tolerance, drought conditions, and flowering. This has been published by Thomas Mitchell-Olds in Arabidopsis, and it was related to the level of expression of FLC. So obviously there is not an immediate direct connection with this, but kind of so it's a speculation, but I would say that probably there's going to be a genetic a trade-off between different responses. And if we were to study different accessions of Arabidopsis and study different levels of SOC1 expression, probably we would find a connection, a link, or an association between SOC1 expression, the level of SOC1 expression, and the level of the degree of drought tolerance. And also, I would like also to study fitness, studying reproductive success that we haven't done.
Okay, thank you. Great talk.
You're welcome.
Okay, is there any more questions for Javier?
Javier.
Yeah.
These genes are conserved in all plant species. So your hypothesis is that this will be the mechanism will be similar in crops.
I think so. So regarding the chromatin regulators, they are very well conserved in all species, from yeast to humans to plants, and the mechanisms, the way they work, it's very similar. The difference between kingdoms and also across species can be about the genes that are regulated. So SOC1 is also well conserved in plants, and probably my guess would be that if our hypothesis is correct, that it's going to hold also in crops. That doesn't mean that it would have to be explored particular responses to particular stresses. That might change a little bit because each crop species is going to have depending on the origin, if it's a tropical species or if it's a temperate species, is going to have a different kind of stress response. But the mechanism probably holds true for crops and Arabidopsis.
Okay, thanks.
Anybody else that wants to ask any questions? Okay, so thank you so much, Javier. And we are gonna go ahead and finish with our last speaker. So our last speaker is Jorge Lozano Juste. He studied biology in Valencia and he did his PhD in the IBMCP under the supervision of Jose Leon, working on nitric oxide signaling. Then he moved to California to the lab of Sean Cutler, where he learned chemical genomics and applied it to identify ABAR receptor agonists in crop plants, especially in maize. During the last year of his postdoc, Jorge was awarded with a Juan de la Cierva and a Marie Curie grant to develop his own project back in Valencia. this time in the lab of Pedro Rodriguez. Since then, Jorge has obtained several grants to continue with his own project of controlling drug tolerance in crops using small molecules. So Jorge, whenever you want to start. I don't think we can hear you. If you can unmute your microphone.
I think now you can hear me.
Yes.
Okay.
Now, yes. Thank you.
Can you see the screen?
Yes.
Okay, perfect. So, well, thank you very much. Thank you very much for the introduction. And also, I would like to acknowledge the organizers for inviting me to share our results with you today. And I have to say that I am enjoying the day a lot. It has been very nice talks, and for sure in the BGP there is a very nice science going on. Well, so in this slide here, I try to summarize the idea of the project. But before I wanna introduce myself, I am Jorge Lozano Juste, and I work in the IBMTP, where I am interested in understanding how plants respond and adapt to stress, and to use this knowledge to develop biotechnological tools to improve plant performance under stress conditions. So in the slide, you can see the resume of the goal of the project, and there in ideal water conditions, plants produce an amount of food, and maize plants are very sensitive to water stress, and the production of these plants is severely reduced upon water stress. So in this project, we wanted to develop small molecules or drugs that we can apply to the plant so we can increase the performance under drought conditions. And to do so, we wanted to follow a chemical approach very similar to the drug discovery process performed in the pharma sector, where first you have to define a target, a protein normally, that is involved in the process that you study, in this case drought. Then you will develop an assay to be able to screen hundreds of thousands of molecules, looking for some molecules able to control the activity of your target. Then all the hits that you found in this screening, you have to perform secondary assays to choose the best hit of the screening that will be called lead. And then normally you need to go through a phase of lead optimization that requires normally the work of organic chemists or medicinal chemists, that they are going to improve the activity of your compound or its solubility or whatsoever. And then if all the steps have been successful, then you will go to preclinical or clinical trials. In the case of agrochemicals, we will go to greenhouse or field trials. So as a target for our project, we choose the ABA receptors. And we choose these targets because we have a lot of information indicating that both chemical and genetic activation of ABA receptors lead to drought tolerance. This is exemplified in this picture here, where you can see two pots that before the experiment, they were very healthy, nice plants. We spray one pot with ABA, and we spray the other pot with a control solution, and we stop watering these plants. And you can see that ABA, the compound ABA, can very nicely protect the plants against drought stress. And this is because ABA closes the stomata of the leaf surface, reducing the transpiration. And this reduction in transpiration, it can be followed or monitored by infrared imaging, detecting the temperature of the leaf surface with thermocameras. You can see here a tomato plant that has been sprayed with a control solution or with a ABA containing solution. And in the plants that have been sprayed with ABA, they have this red color that indicates the temperature is higher than the control-treated plants. And this is because the ABA-treated plants have closed the stomata, so the transpiration is reduced, and there is less evapotranspiration, so the temperature of the leaves are higher. But not only we can activate drought tolerance chemically activating the receptors, but also genetically. And this is also, let me check. This is also exemplified here in this figure. These are Arabidopsis plants that overexpress the PIL4 ABA receptor, and they are much more drought tolerant than the wild type plants. And again, we can follow this drought tolerance by infrared imaging, detecting the increase in temperature of the plants overexpressing the receptors over the wild-type plants. And the peer-pill receptors or the peer-pill ABA receptors are nice targets also because you can translate the findings into crop plants. That is very important. And here you can see an example of that where wheat plants have been transformed with the peer for wheat ABA receptor, and these transgenic plants are much more drought tolerant than the wild. So a little bit more of introduction about the ABA signaling. In the left-hand side here, you can see that under normal conditions, the PP2C phosphatases are interacting and inhibiting the SNRK2 kinases and keeping the ABA signaling off. But when drought arrives, there is an increase in the synthesis of ABA and in the transport of ABA. So now the receptors bind to the ABA, they change its conformation. So now they can interact and inhibit PP2C phosphatases. So the SNRK2 kinases now are free to autophosphorylate and to phosphorylate downstream signaling factors, activating the ABA signaling pathway. So in this project, what we proposed, it was to apply this ligand-induced protein-protein interaction that we just explained between the peer-pill receptors and the PP2C phosphatases, and establish a GIS2 hybrid-based screening to look for molecules able to activate maize ABA receptors. So the first thing we did is to identify maize ABA receptors by bioinformatic analysis. We have a dendrogram here, a phylogenetic tree here, so in the alignment of maize receptors and Arabidopsis receptors. And maize receptors group very well in 3 different clades as the Arabidopsis ones do. So what we did is to pick one representative member of each of these clades. In this case, we pick 2 proteins. We clone them in the GIS2 hybrid vectors, and we perform the chemical screening of a 20K chemical library. So we did 4 different screenings in parallel to screen this large chemical library. And cutting a long story short, here are the 8 hits that we found in our screening. These are the names of them. And this is a GIS2 hybrid panel. You can see blue, there is interaction, white, there is no interaction. Here you can see all the maize ABA receptors. There are 12 ABA receptors in maize. All of them are activated by ABA, as you can see here. And the previously identified ABA receptor agonists, like quinabactin, can activate only, in this case, PIL1, PIL2, and PIL3, while our hits can also activate other receptors here and there that are not activated by previously identified ABA receptor agonists. And this is more evident if we analyze it in the Arabidopsis ABA receptors. Again, here you have the receptors. ABA activate all of them again. Quinabactin activates only PIL1, PIL2, and PIL3, while our compounds, or the compounds that we found in this screening, are able to activate many more receptors than the previously identified agonist. So our compounds seems that they have expanded the repertoire of the receptors that they can bind to. So now we have 8 molecules that are interesting. We need to know which one is the best one. So to do so, we produce the ABA receptors of maize as recombinant proteins to do a lot of in vitro assays that are summarized in this table. Here are the ABA receptors. These are the names of the compounds that we have found in the screening, and here you can see the intensity of the binding of each molecule. So, in conclusion, this molecule, the 72C10 compound, it was the best hit that we got in our screening, and we will work with that compound from now on. The first thing, well, I show you here. This is the structure of 72C10, and we perform some structure activity relationship studies to determine which part of the molecules are more important. And we found that this part of the molecule is very important, but when our collaborators, the organic chemists, saw this compound, they quickly suggest us to get rid of this group here, this nitro group, because they understood that this could be very reactive. So they proposed to change it, and they changed it by a nitrile group here that has some of the By the other hand, it has some of the properties that it has also this group here. So we changed the structure of 72C10 and synthesized ceabactin that has this nitric group here. And the next thing to do was to see if this compound is bioactive in maize. And for that, we developed some assays to reduce the amount of product that we need to use in order to see if the compound is active in maize. So with this, we developed this kind of assays where you can follow two things. First thing you can follow is the reduction of the coleoptile length with increasing concentrations of ABA. And the other thing you can follow with these assays is the reduction also on the root length with increased ABA concentrations. So we test our compounds in this kind of assays. We check root length by one hand and coleoptile length by the other hand. And we saw that ABA can reduce root length. This is not new. But surprisingly, quinabactin was not able to reduce root length in maize. It seemed that quinabactin was not active in this kind of experiment. But our compounds 72C10 and ceabactin both nicely reduce root length in maize, being these compounds bioactive, and perhaps ceabactin is a little bit more active than 72C10. The same happened in the coleoptile length experiments. ABA could reduce nicely the length of the coleoptile, while quinabactin again didn't show any activity on these assays. And 72C10 and ceabactin both were able to reduce coleoptile length as ABA does. So these compounds were active in maize, but what we wanted to know, what we were more interested in, it was to see if this compound can reduce transpiration. And for that, we contacted and we could establish a collaboration with Sergio Neubauer from the Universidad Politécnica de Valencia. And he helped us a lot to determine the stomatal conductance of maize plants treated with ABA or with ceabactin using infrared gas analyzer, an infrared gas analyzer. So we could prove, or we could see that it was the case, ceabactin can actually reduce the stomatal conductance of maize plants. But when we studied this transpiration phenotype more in detail using infrared imaging, we saw that, as expected, ABA could increase the temperature of maize-treated plants compared to the control here. But in this experiment, ceabactin or 72C10 didn't show a clear change in temperature. So what this means is like this, the ceabactin for sure it can reduce stomatal conductance, but this reduction is not enough to reduce the transpiration significantly. Okay, this was also confirmed when we did an RNA-seq experiment to see what was the molecular phenotype of plants treated with ceabactin. And in this case, we treat maize plants with ABA or ceabactin, and the results, the conclusion of the results is that 90%, around 90% of the ceabactin regulated genes are also regulated by ABA, meaning that ceabactin is specific. So ceabactin only activates ABA response, but by the other hand, only 11% of the genes that are regulated by ABA are also regulated by ceabactin. I mean that ceabactin is very specific, but it's a weak agonist. It has a weak effect. So where do we go from here? No? What do we do now? So what do we do is to improve the activity of the compound, and to do so we choose a path, and we choose to solve the structure of the ternary complex of the PIL1 receptor here in brown, with the ceabactin in purple, and the PB2C4 fatase HAP1. So we wanted to use this structure to guide the optimization of our ligand. So what we wanted to do is to check which amino acid contacts are between, or which contacts are between the molecule and the amino acids in the binding pocket to increase these contacts and make them more strong. So we did this work in collaboration with the lab of Armando Albert and Lourdes Infantes, and they not only solved the structure of PIL1 in complex with ceabactin and HAP1, but also the structure of PIL2 with ceabactin and HAP1. And the first thing that we saw, to our surprise, is that the pose that ceabactin has in PIL1, you can see it here, is different from the pose that it acquires in PIL2. And this is in agreement with the activity of this compound, with the activity of ceabactin in PIL1 and in PIL2. And the in vitro activity of ceabactin in PIL2 is lower than the activity on PIL1. And you can see how the molecule has a different arrangement in the pocket of pill 1 here in blue, or in the pocket of pill 2 here in pink. It seems that the compound has moved like 60 degrees or so in the axis. And we know that the correct position is the position that the molecule has in pill 1, because when we overlap with the structure of pill 1 and ABA, you see this very nice overlap between ABA here in white and ceabactin in blue. So you can see that there is a very nice overlap of this part of the molecule. Here is the ketone of ABA, super important for binding to the pocket, and this methyl group here, the C6 carbon, that is also very, very important. So well, what we wanted to do was to improve the activity of our compound, and we focus our attention in one region of the receptor that has been described as a hotspot to improve the activity of some ligands. This region is this cavity that you can see here. It's kind of a hole. You can see the ABA inside the pocket here, and it's called the 3' tunnel that is magnified in here. So what it has been described is like if you can make your ligand to extend through this tunnel, you can increase the activity of your compound. So what we did first, we checked in our crystal structure if ceabactin was entering the tunnel, and you can see here in pink ceabactin is inside the pocket, but it's not going through this tunnel, this window here. So we designed 2 chemical modifications that we thought they could increase the activity of the compound because it will go through this tunnel. So one was to add a cyclopropyl moiety in meta position. This is why we call it ceabactin M. And the other one, second molecule, it was a cyclopropyl in the ortho position. So this work we did in collaboration with Juan Carlos Estevez in the Universidad de Santiago de Compostela, synthesized both compounds, and we even did the docking predictions, and the docking predictions predicted that actually the modification of ceabactin in meta position will make that this cyclopropyl will go through the tunnel, increasing the interactions with these hydrophobic amino acids that are in this pocket. So this was actually the case when we tested this compound in vitro. We found that, well, this is the activity, the IC50 values of ceabactin M. This is the IC50 values of the ceabactin, the parent molecule. So there is a 10-fold increase in potency in vitro here with this receptor, and a 2 times increase in potency over this receptor here. And this increase in potency in vitro, it's also, it goes with an increase in the activity in vivo as well. You see here ABA inhibits germination, right? So ceabactin M can inhibit seed germination at 1 micromolar, while the parent molecule, ceabactin, can inhibit germination only at 25 micromolar. So this is an 25-fold increase of activity in vivo with respect of the parent molecule. We have increased activity of our compound. And importantly, we wanted to see if this compound can reduce transpiration in plants. So we call it, with this ceabactin M compound, we call it ceabactin 2.0. And now you can see that it very nicely can increase the temperature of the plants after spraying with the compound. The effect is not as strong as when you treat with ABA, but you can reduce the transpiration of these plants. These plants here in this thermograph are not maize. These plants are Setaria viridis. It's a C4 monocot model for plants like maize or sorghum or sugarcane that I have been working a lot in the lab, and I will be very happy to go back to you and tell you the Setaria story another day. We want now, of course, to not only check that this compound works in Setaria, but also we want to check if this compound works in maize. Anyway, and because this approach works so nicely for us, we wrote another project. We contact with a company that are specialized in organic synthesis, and together we got a Retos Collaboration grant to improve the molecules that we have obtained in several screenings. So we started with the scale with ceabactin 2.0, but also with other 2 molecules that we found in another screening campaigns. So with the structure of the compound and the receptor, and with the help of the organic chemists, we have synthesized more than 19 new ABA receptor agonists. And among them, I want to highlight one of them, probably the one that I'm more happy with, that is this S1A017. And this compound is better than the previous one, that's Avactin. But if you pay attention, you can see that it's around 4 times more potent than the endogenous ABA molecule. So now we have synthesized, we have designed and synthesized a molecule that is even more potent than ABA in vitro. And these in vitro values are also very well extrapolated to in vivo data, because here we didn't try maize yet. We are looking forward to do that. But we tried in Nicotiana bentamiana. These are Nicotiana bentamiana plants that have been sprayed with a control treatment with 100 micromolar ABA, with 200 micromolar ABA, or with 100 micromolar of our compound. So you can see that even with half of the concentration, our compound is able to produce a stronger effect in transpiration than ABA. So as I told you, we are looking forward to perform these experiments in crops, especially in maize, to kind of round out this set of data. And we think we have a very nice ABA receptor agonist with potent activity. And with that, I hope I have not been very quick, but I want to finish my presentation here, acknowledging all the people involved in this project. I mean, it's a project that involves people working in plant biology, in structural biology, also organic chemistry. So there are many people involved, but I would like to especially thank Pedro Rodriguez for hosting me in his lab during these years, the group of Armando, Albert, and Lourdes Infantes for the structural biology, Sergio for the physiological measurements, and Juan Carlos from Universidad de Santiago Compostela and Alquimia for the expertise in the organic synthesis. And well, I would like also to acknowledge the funding sources and to all of you for your attention. And now I am open to questions if you have any. Thank you.
Okay, thank you, Jorge, for your presentation. If there is any questions for Jorge, please go ahead and unmute your microphones.
I got a question. So okay. Yeah, I was wondering. So ABA is known to prime plants in order to increase tolerance to diverse abiotic stress, not only drought. Have you checked that? How you check if your compound increases tolerance to drought, but also to cold or to other abiotic stress responses?
Okay, it's a good question. I didn't try them. It's very laborious to do the drought experiment, so we are focused only on this kind of stress for now. But it's true that ABA is related also to salt stress, to cold stress, to other stresses. So there are options that our compounds could work also in this kind of stresses. But answering your question, I didn't try other stresses other than drought.
And just another one. How about the stability of this compound? So it would be possible if it's stable in field conditions, it's going to be able to be there to provide a higher tolerance, if that's the case, and also if it could also produce some kind of memory that it's going to last in field conditions.
Okay, so answering your first question about the stability, we don't have a reason to think that they are not stable under field conditions, but it's something we didn't try. It's something we didn't test yet. So when we do the experiments or the how to say, what our scheme of working or our way of working is that we do a screening of molecules. We find some that can bind to the receptors. We test the effect of these receptors in greenhouse experiments, but this is the farthest we can go. So from there, our plan is, so we patent the molecules, and then if there are some companies that are interested in the compound and to check if it works in the field or it works or they want to improve the preparation or the formulation or whatever is their tasks. We don't go so far, but well, we would be interested to know if these compounds are stable or not. Yes. And then the second question was the memory. The memory, we didn't do an experiment to test that. And actually, I will have to talk to you or with someone that is expert in the field to know how to do these memory experiments.
Mm-hmm. Okay, thank you.
Thank you.
Jorge, this is Antonio Molina.
Hello.
Nice talk, as I mentioned already to you a few days ago, okay? Regarding the other talk. So I have two questions. The first one is related to the fitness of associated to this compound, because you showed that in some of your experiment, when you treat with EVA, particularly in the seedling state, you have a reduction of the hypocotyl and also from the root length. So how is this new fascinating compound comparing with the EVA in this fitness cost?
Okay, so these new compounds are still very active in this kind of experiments. So you get reduction of root growth, you get reduction of coleoptile. Of course, these are just experiments to test the bioactivity of these compounds, right? It's not the final use what we are testing there. So when we do this small assays to check coleoptile length or root length, well, this new compound is very active. I have to say that it's not, in this case, it's not more active than AVA. You saw that when I show you the thermoimaging, that it seemed that the compound, it was activating or it was reducing transpiration more than AVA itself. But in the root growth, coleoptile growth experiments, they are same as AVA. It doesn't go further. And then for the final use, of course, you are going to have a trade-off when you apply an AVA receptor agonist into the plant. You are going to reduce transpiration, so the plant is going to grow less. But by the other hand, it's going to keep the water in the substrate. No, it's going to do this water banking thing that is going to let it survive to the drought in the future.
Okay, so the second question is, yes, now based on your experience, it may be feasible just to use a different approach, just starting from your docking analysis with the natural compound EVA, and based on this to try to improve, I mean, to do something better than the nature, instead to go through different rounds of screening, or this is not possible?
Yeah, there are some people that have tried that. There are two groups in Japan that have tried to modify the AVA molecule itself, right? To make it like some changes to make it more potent. It seems that the AVA molecule is ideal there. So any change you do in the molecule, it doesn't help with the activity. Plus the chemistry, organic chemistry of AVA is kind of tricky. I'm not an expert on organic chemistry at all, but is what the organic chemist says, that is something very, very hard to work with. So the people that have been working on that, they have been appending like another aromatic rings into the AVA ring already, or to changing the carboxylic acid as well, but this doesn't improve the activity of the compounds. They have actually checked a lot of AVA-related metabolites, and they are not as active as AVA. As a strategy, to find potent compounds, I would recommend always to do a screening phase to find probably a novel scaffold, if you are lucky, novel chemical features, and then from there you go and you improve it by organic chemistry.
Okay, thanks.
Thank you.
Okay, thank you, Antonio. Is there any more questions for Jorge?
Yeah, I have a question. Here you are showing that you're looking for a molecule that acts as AVA, but I guess that with more potency. But have you looked for any other feature that can be like additional to AVA, like something that AVA does not, but this molecule could do, like?
Yeah. So in the case of the latest molecule we have found within this last project with the company, we didn't go so far yet. But with the Seabactin 2.0, for example, or with Seabactin, and indeed, what we did is this RNA-seq experiment, right? So with this RNA-seq experiment, we see that like 90% of the genes that are regulated by this molecule are genes that are regulated by ABA as well. So nothing new there. And then actually when we use genetics and when we use, for example, like sextuple mutants or on the ABA receptors and we apply this molecule, there is no effect at all. So I think they are very specific activating ABA receptors and they don't seem to do much more than that. Okay. I hope I answer your question.
Okay, thank you.
Okay, thank you, Bruno. We have a little more time for one more question, if there is somebody else.
I have been very quick, right?
Has been okay, thank you.